Professor Oyama is an assistant professor of biology at McGill University, where she's been since 2017. Tomoko did her PhD in Hugo Bellin's lab at Baylor College, and then a postdoc under Marta Zlatic at Janelia. And so she'll be talking today about some of the work she's done on the optical and genetic approaches to understanding escape behaviors in Drosophila. And so, Tomoko, the floor is yours.
Right. Can you hear?
Yes.
All right. Thank you very much for the introductions and then the invite that give me an opportunity to talk about our research. It is okay?
Yes.
Sorry. All right. So I will start from this cartoon. This is my favorite cartoon, Hoops and hoping that he actually right now need to do something, right, based on the how much the tiger is mad, angry, that he need to calculate the information, basically facial expression of the tigers or shaking, how much shaking his hands, right? Then probably he has an experience that he need to do what he should do next. But actually, he's not doing this job. His brain collects the information, then produce the actions. The brain is a main function of brain is this computation is all the time. And all the animals doing this job. And my favorite animal, Drosophila larvae, also doing this job every moment. And this brain, the small larva brain has several benefits to use this use this brain, right? Because one of the reasons is we have an entire nervous system of EM images, which allows us to generate the connectome map in the synapse levels. So currently we have 70% of the neurons are reconstructed. Then we know the overview of the connectivity in this nervous system. Not completely, but we know the big view of the connectome map at the synapse levels. As well as we have a lot of genetic tools allows us to manipulate the single neuron levels and then monitor the neural activity from the entire brains. And today I will not talk about this entire brain imaging like zebrafish, of which previous talk, but we can do the similar stuff in this context. This is a light sheet microscope images of the entire nervous system, about 5 hertz imaging of the entire nervous system, and with the optogenetic activation. We are activating the single segment, then monitor the neuron activities. It looks like forward wave behavior we can observe. Then how about the behaviors? Now behavior is coming out, I hope. Right. So this panel is showing that we activate the 5 different neuron types, then observe the behaviors. How much different behavior can be detected by the computationally? That statistically different behavior we observe 29 different, suggesting that even this maggot, tiny, simple, just crawling and eating and then become pupa. And then they just need to eat all the time and then become pupa, that's their job. But still they can produce 29 different, statistically different type of the behavior, suggesting that we can study how this network, the neurons, can generate a different type of the behavior. That is our questions. And then I would like to show that this EM pictures, how it works, is that this is a single view of the section of the EM that we can identify the synapse, shown here, and then synaptic partners. Because of we have a series of the sections of the EM image of this one, but then we can reconstruct the neurons manually or automatically, then a synaptic partner can be identified. So this is, for example, white, pink, and green are sensory neuron types, neurons, then we can identify synaptic partners. So then how much stereotyped between the animal to animal? That is another challenging because of EM sections images is only one or few, right? But we need to tackle the different animals. But insect neurons are stereotyped. That's another benefit to use this one. So for example, this is the EM, the neuron reconstruction shown here. Then based on the cell body positions, then neurons, the others, we can identify the same neurons, single neurons from the light microscope images shown here. Then we can manipulate this neuron using the genetic tools. And we know that about this animal has 10,000 neuron types. Mushroom body, which has a function for the learning, that connectome is individually different. However, other neurons, for example, ventral nerve cord of the neurons are stereotyped so that we can really manipulate the single neuron types in the different animal to animals. Okay, so today I would like to talk about how we are tackling the identify the circuit using the single neuron levels. Okay, then we are interested in the escape response, rolling, and fast crawling. Okay, this is another. These actions are mutually exclusive, so they cannot do these actions both at the same time. So they need to choose which one is first, which one is later. Okay, then rolling behavior is observed by attacking by the wasp. This is a parasite wasp, so that they need to escape from the parasite wasp. But right now, I'm evoking rolling behavior using the optogenetically. So without wasp, we can evoke rolling behavior. And the first crawling was observed mild aversive cue or after the rolling behaviors. Then I already mapped, our lab already mapped out that this rolling circuit, EM and behavior and neural imaging of the functional connectome that's shown here. Basically, sensory informations are integrated in the first order interneuron called Bayesian cells. And this Bayesian cell connects the ascending neuron or locally to the goal neuron, which is a single pair of the neuron to evoke rolling behaviors. Okay. Then this ascending neuron actually coming back to the goal neurons so they can modify these behaviors. The simple escape response actually, it's very complicated circuit they have. Now, this is a pinching. It's not optogenetically, but you can see after pinch, first they roll, then crawling away, right? They need to first roll, then crawling away. This is the histogram of the actions that you can see the color lines is crawling and the black line showing the rolling behaviors. That first they roll, then crawl fast. Okay, this is a sequence of the actions to generate how actually brain can generate this action sequence. That is our simple questions. All right, so one model has been proposed that in the zebrafish vocalization that one action evokes second action evokes third. But we think that another model is hierarchical suppression model, which means that each action has a module and inhibit each other's. And the one action first, dominant, need to be inhibit the second and somehow decrease the first action and the second action need to be appeared. But neural substrate of this, the both model has not been still unclear. Then we can maybe understand this one in the simple escape models. And actually first order interneuron, when I activate optogenetically, we see the sequence of the behavior still, rolled followed by the fast crawling. This is a behavior sequence of the actions. But however, when we activate the command neurons, we only see the rolling, but not followed fast crawling, suggesting that we think that this behavior is hierarchical suppression model, means the rolling behavior somehow inhibit the fast crawling. So we want then that somehow they need to terminate the fast loading behavior to appear the fast scrolling, right? And then what's the inhibitory mechanism of this sequence action? That's our questions. To find the neurons that evoke this shifting to the fast scrolling, we have taken the forward genetic approach. Basically, we activate the first order interneurons optogenetically with the X neuron, also optogenetically, then find the reduced rolling or increase the fast scrolling or decrease fast scrolling, somehow defect. So then we have lots of GAL4 lines, single neuron types. This is a catalog of the example of the GAL4 line to manipulate the neurons. Then basically we activate the first order neuron, evoke, roll, follow the fast scrolling, and then X neuron to find some defect. Then basically we found one neuron types called to the SS4185 neurons. So the shadow represents the optogenetic activation path. Then we can see the reduction of the rolling when I activate together with the Bayesian cells. And the probability of the rolling shown here, you can reduce the rolling. This is the behavior. Then how about the fast scrolling? We see the shift to the little bit reduce the fast scrolling, but also timing of the fast scrolling much earlier. This suggests, this is a complement or supports the idea that higher ACCO suppression model, which means the rolling inhibits the fast scrolling, right? Fast scrolling timing is not controlled by the first order interneurons. Okay. Then we inhibit these neuron types, that we see the increase of the rolling when we inhibit these neurons. However, the probability of the rolling entire time, if you look at animals, we do not see the reduction of the rolling behaviors. What's going on, right? It looks time series increase the rolling, but probability is not rolling. And then we look the duration of the rolling of each event. We see the increase of the rolling durations, suggesting that this neuron seems like important for controlling the rolling durations. Now, which neuron is? This is the lines of the neuron types which manipulate, we found that this is the mushroom, the top brain part. Maybe I need to put the laser point, sorry. This one is a mushroom body, which is important for the learning and memories. And we know that this neuron type, mushroom body neuron, are not involved in the rolling behaviors. We have tested only this neuron activate or inhibit. We do not see any phenotype. So we think that these descending neurons are important for the controlling this duration of the roll. And actually that we stain the neurotransmitter of this neuron that we know the GABAergic. So seems like an inhibitory. Now we can go back to the EM connectome. And we found these neuron types. The first, I would like to show that this ascending circuit is so the Bayesian cell, which can evoke rolls followed by the fast scrolling. Then this neuron are connect to the A00C neuron, ascending neuron, which I showed before a little bit briefly, beginning. Then this A00C neuron are actually connect to the sending to the information to this SEIN129, already named. It's luckily we already reconstructed these neurons. Then the descending neuron sending back to the Bayesian cell as well as A00C. So we saw the very nice feedback motif in this small circuit. Then might be that this is EM connectome. We don't know this is really functional or not. Then we need to test we would like to test the functional connectome of this circuit. So basically we activate the Bayesian cell using the crimson, which is red shifted channel rhodopsin. And then simultaneously we image the GCAMP in the A00C neurons. So basically setup is like this. We put the two-photon microscope under the microscope, we put the brain, then we can activate the crimson using the red LED through the objective, then simultaneously the neural activity using the GCAM. Then this is the example of the, sorry, I hope laser doesn't work. Sorry. Last time also didn't work. I'm sorry. I test today, but maybe automatic. Then, okay, it worked. So you can see that the square showing the when activation comes, nicely, reliably, we can see the activity at the axon terminals or dendrite. In fact, I tried to the cell body recording. We also tried to the cell body recording much slower, as well as some neurons, we do not see any activities. It really depends on the neurons that we found. Okay. Then we can change the intensity of the stimulation as well as 10 Hertz or 1 Hertz shown here. You can see that the nice response, dose response curve of the response in the A00C neurons. 10 Hertz, we see the already 10 microwatt stimulation already saturate of the activity at the 10 Hertz. Right. Then how about that this descending neuron named SE1N129? We see that more slower and smaller response to the same stimulations. And then especially we are interested into this longer activity compared to the A00C, suggesting that maybe this dynamic of the activity is enhancing to the termination of the action to evoke to the next actions. Okay, so now this the neurophotonic teams, we would like to evaluate the GCAM8 using the similar setup. That this is coming from the Janelia Research Campus data from the website. They already tested the GCAM8 at the neuromuscular junctions. So they stimulate the motor neuron using from the motor neurons and then observe the motor neurons activity using the motor neuron activations. And then you can see, as you can see, that GCAM-F seems like very fast, nice response. 8 and S is a slower, but the much higher delta F over F they found. And then with this, our group is still small for currently for 3 to 4 graduate students, and I'm still looking for the peoples. So if you are interested, please come to us a small brain if you would like to do a small brain work. And then also I would like to thank you to all the funding source. Thank you.
Thank you very much, Tomoko. It's very interesting. So again, if there are any questions, please go ahead and either raise your hand, post them in the question box. I actually had a question for you, Tomoko. Many of the observations that we're trying to relate the anatomical structure of connectivity and the synaptic connectivity to the behavior are sort of depending on the idea of a direct monosynaptic kind of connection or direct synaptic connectivity rather than diffuse release into, of a neuromodulatory substance, for example. Do you think that there's the possibility of especially in something like this, where a behavioral state is moved from one state to another for some kind of volume release?
So we do not know the neuromodulators, how affect this hardwired synaptic connectome, but that's we already know the many neuromodulator neurons in these systems, So we can map out to the layout to the connectome map. Then try to understand, but still, it's a next question.
I guess the nice thing is sort of like what Antoine and Vincent are trying to do with post hoc labeling. You already know the identities of the cells, and so you can kind of do that pre-hoc.
Right.
Nice. Any other questions from the audience? Wait a minute or two.
Well, maybe I can ask one.
Thanks, Martin.
I'm just wondering, what's the strategy that you use to specifically target different neural type in this model? You know, the opsin you use and calcium sensor. You seem to target a specific group of neurons. So how do you do that?
So we have a GAL4US system that only specific neuron we can express the crimson or some other chronos or opsins or inhibitory. We use a KIA channel or tetanus toxin, but basically US GAL4 and then Lexe up, which has a differently. So we can really 2 or 3 effectors can be manipulated in the different neurons. So it's very powerful, I think.
Yeah, indeed. Thank you. Very nice talk.
Okay. If there are no other questions, there is a question. So Vincent Breton-Prevencher says, do you have an idea how linear is the relationship between the number of action potentials and the calcium response for the different calcium proteins that you've used?
Sorry, can you repeat again? I'm so sorry.
Sure. Actually, if Vincent wants to ask the question, or you want to unsilence yourself, I don't know if that's possible. Maybe I'll ask. So it says, do you have an idea how linear is the relationship between number of action potentials and calcium response for the different calcium proteins that you've used?
Right. So, okay. I personally do not recording simultaneously physiological recording and imaging, but I think we could compare this neuromuscular junction recording from the Janelia one, that lots of already recording of the neuromuscular junctions, I think physiological recording. Then we can match up to we can compare the at the neuromuscular junction, physiology data and imaging. That's possible.
Oh, thanks. Yeah, I mean, it's interesting. We have a lot of different calcium indicators now available with different kinetics. And we were talking with Jean-Claude Bake the other day. He suggested it might be that for certain cells with fast dynamics that we need to use different calcium indicators than for other cells with slower dynamics. I think it's an interesting possibility that we'll have to customize the indicator for the cell type.
Yeah, if I can add to this, Ed, and I think it's really important to do it in real world situation, because there's a lot of discussion about how nonlinear some of these calcium sensors are and blah, blah, blah. But we've personally found in some of our paradigms in vivo where we control spiking that we have actually in the end a very, very linear relationship between number of spikes and the response. So I think, again, it speaks to the importance of testing some of these in real tissue and so on, because there's so many, it's so multifactorial sometimes what results in the response in the end, that what we think is happening based on the in vitro data or the simple properties of the indicator does not necessarily translate.
Okay, if there are no more questions, I think we can close the session by thanking the speakers. I think there was a really nice theme here of structure and function. So thank you both, for all three of you, for these presentations. They're great.
Okay, so it's 1:15. I think we should get started just to stay on time. thank you everyone for being here. I just want to thank Yves and his team, as well as Mario, for really putting this great symposium together. It's really a wonderful opportunity to be able to see what everybody else in the community of neurophotonics is doing. So thank you, and thank you to all our participants. I'm biased, but I think this is the most important session of the day and the most interesting one. So it's great that you're all here with us. I had prepared like about a dozen slides of introduction for you, but Mario said we have to be on time. So just quickly introduce the speakers and move on. So I'm very delighted that we have two speakers from the labs of Tom Durkin and Ed Fawn at the MNI, who are going to tell us about their findings. And then Pierre Marquet from University Laval. So again, thank you all for, thank you to our speakers. We're going to start with V. Mohammed, who's going to talk about microfabricated disc technology. So her talk is going to last for about 15 minutes, and it's going to be followed by Ghislaine Deyap, who's going to talk about characterizing patterns of neural activity in midbrain organoids, both from the Montreal Neurological Institute. And because of timing, we may not take any questions after their presentation and move on straight to the presentation of Pierre Marquet, who's going to be for half an hour. And then, I mean, hopefully not the full half hour, but then we'll start taking questions from everybody, okay? And Pierre Marquet is going to talk to us about holographic microscopy. So without further ado, we're going to start with V. Mohammed, who's a postdoc in the group of Ted Fawn. V, the floor is yours.
Thank you. Try to share my screen first. Do you see the slides?
Yes.
Yes, perfect. So thank you for the invitation. The data I will present you today is about a rapid scale-up in human midbrain organoid generation. So my talk will be followed by Ghislaine's one, who will dig into the electrical activity recorded from those human midbrain organoids. So studying Parkinson's disease in the lab presents many challenges, the main one being the limitation to access tissue from living affected patient. And so the emergence of induced pluripotent stem cells called iPS allow for the differentiation of neurons in vitro in a dish from a blood or a skin sample. However, to reproduce the complexity of the brain's environment, researchers are developing more refined iPS-based tools to model better Parkinson's pathophysiology and advanced drug discovery. And so recently, the development of cerebral organoid is a major technological advance in the stem cell field, and Madeleine Lancaster successfully generated in 2013 this tissue that contains many features of the brain. So cerebral organoids are composed of mixed population, including neurons, astrocytes, and oligodendrocytes, which are self-organized in the space, and they also exhibit cerebral function like saltatory transmission, and so they do have node of Ranvier and synapses. 2013, different type of brain organoids have been generated based on adaptation of this initial protocol published by Dr. Lancaster. And so now the modulation of specific inductive factors gives rise to multiple types of brain organoids that can be used to model neurodevelopmental and neurodegenerative disorders that affect distinct regions within the brain. So for example, midbrain organoids to study Parkinson's disease. I used to produce organoids with 96 well plate ultra low attachment and maintain them in culture on orbital shakers with 6 well plates stacked on each other. This technique is now used extensively in laboratories, but it has many disadvantages. It does require a lot of pipetting, of course, with the 96 well plate formats, single embedding with matrix scaffold in each well to provide an extracellular scaffold to the midbrain to grow, as well as single transfer of the midbrain organoids into final plates. From a maintenance perspective, media changes occurred 3 times per week. So I'll let you imagine the time that is required when you have to handle hundreds of plates stacked on several orbital shakers. And for all these reasons, I refined the process of midbrain organoids generation, and that led to a more efficient workflow to generate and maintain those midbrain organoids for long-term culture. The paper is actually in press, and my aim is to ameliorate the organoid production, making it robust, consistent, and scalable to levels where organoids can be provided to users across the life science sectors for research and drug discovery. In this manuscript, I describe a novel protocol using a microfabricated like embedded body device called EB-Disc to generate large batches of homogeneous midbrain organoid across multiple IPS lines. So I derive midbrain from different IPS lines using classical midbrain partitioning factors, and to generate those midbrains from a single EB-Disc containing 948 microwells, I started from IPS colonies at 80% confluency. The process to generate those midbrain takes about 8 days. After dissociation of the IPS colonies into a single cell suspension, I seeded the cells on top of one of those EB-Discs and centrifuged the device to form one cellular aggregate within each microwell. The subsequent step consists of driving the cells toward a midbrain fate with several media changes containing the differentiation factor of interest in stationary culture, until the midbrain organoids were embedded directly on the EB-Disc at day 7 in a matricial scaffold to promote the tissue development. Finally, on day 8, the disc was lifted up and flipped to a dish containing final differentiation media, and the matrigel embedded midbrain were then transferred into bioreactors containing final differentiation media to promote the maturation of the midbrain and maintain them in culture prior to the collection for experiments. So the generation process described to scale up the production of midbrain was based on the use of this microfabricated EB-Disc. For the production of batches with several hundreds midbrain, I use EB-Disc with 948 microwells in partnership with Inuvio, which is a Montreal-based startup. And the EB-Disc are pre-coated with an ultra-low attachment coating made of pluronic acid and with defined dimension as depicted here on the lateral section and detailed here in the text. So the discs were made from PDMS, a flexible material permitting easy handling of the platform for midbrain production while being biologically compatible with the generation of the neuronal tissue. So I observed the progressive appearance of aggregates within the disc from the seeding day, D0, until they transfer into the bioreactor on day 8. Indeed, after 48 hours in the disc, EB forms and present smooth edges. They grew and start to present with neuroectodermal bud extrusion on day 7 after the midbrain partitioning media. And the embedding with matrigel scaffold on day 7 led to even more pronounced bud extrusion 24 hours later. So I quantified the area of EB and midbrain organoid derived from 5 distinct IPS lines, including two Parkinson disease lines and compare the size from day 2 to day 8 within the disk using an in-house cell profiler pipeline that creates corner mask to visually verify the area to be quantified. So our analysis demonstrates that EB from all lines grew in size from day 2 to day 8 within the disk to attain a diameter of about 500 to 600 micrometer. The area of the midbrain at day 8 from the disk were similar across all lines. This is what you can visually observe from freshly transferred midbrain compared to more mature one. So I used several disks containing 948 microwells in parallel across different lines that were synchronized in confluency to generate batches with different conditions. So the aggregates in each disk were transferred on day 8 into a specific bioreactor and placed on a magnetic stirrer housed within an incubator. Each magnetic stirrer can hold up to 9 bioreactors, meaning that 8,000 organoids are growing simultaneously in this setup, approximately. Additionally, to assess the diversity of cell population within the midbrain, I stand cryosection for S100 beta, MAP2, and TH, marker for astrocytes, neurons, and catecholaminergic, dopaminergic neurons, respectively. In addition to immunochemistry, I perform quantitative PCR to detect several transcript nodes to be expressed in the midbrain, including, for example, Nor1, LMX1B, A, TH, COMT, or DDC. And so they were present in the midbrain tissue and absent in the parental IPS line. Additionally, after 100 days in final differentiation media, midbrains start to develop and accumulate spontaneous neuromelanin granules. This observation was previously confirmed to be a feature of functional dopaminergic neurons. Next, to further validate the midbrain identity and the reproducibility of our method, Dr. Maria Lacalle cleared and stained with a method called Cubic 4 6-month-old midbrain from our control line for TH, the marker of the dopaminergic neurons, and MAP2. She observed the presence of dopaminergic neurons, stained for TH, within the HMO as expected. So I will let you appreciate the beauty of this 3D staining and the reconstruction that Maria Lacalle did. And so the idea is to avoid a cryosection and to be able to have a better visualization of the entire 3D structure. So finally, the volumetric quantification of MAP2 and TH staining percentage showed that similar levels of neurons and dopaminergic neurons were present across each of the 4 midbrain organoids tested. And the TH over MAP2 ratio showed similar yields of dopaminergic neuron in 4 midbrain organoids. So taken together, our findings indicate that large numbers of midbrain can be generated with this protocol, and they have a midbrain identity and similar cell type relative to each other in the tissue. So I would like to thank the NeuroEDDU and especially Dr. Fun and Dr. Durkin, as well as the partner for the fundings. So now I'm going to let Ghislaine take over the presentation and talk about more the electrical activity within the midbrain organoids. This is your turn, Ghislaine.
Thank you, Reed. Okay, so hi everyone. Thank you for having me here today. Today I'll be presenting a little bit about my project titled Characterizing Patterns of Neural Activity in Midbrain Organoids as a Model for Studying Parkinson's Disease. So Parkinson's is the second most common neurodegenerative disorder after Alzheimer's, affecting approximately 1% of the population. It's characterized by progressive loss of dopaminergic neurons in the substantia nigra pars compacta, along with these aggregates of alpha-synuclein forming Lewy bodies, as well as an increase of oligomeric forms of alpha-synuclein, which contribute to neurotoxic effects of Parkinson's by playing a role in things like mitochondrial defects, inflammatory responses, and lysosomal dysfunction. And so this all leads to the progressive loss of dopaminergic neurons, leading to both cognitive and motor decline. So next, I'll just quickly introduce iPSCs and neural organoids as a model for studying neurodegeneration. So we can take samples of human skin cells, blood, or urine, and reprogram them into induced pluripotent stem cells, or iPSCs, which can then be further differentiated into specific 2D cultures, such as dopaminergic neurons, motor neurons, or microglia. Or they can be used to make 3D cultures, such as neural organoids, which develops a more heterogeneous population of neurons and cell types, as was just mentioned by V. And so using stem cells can be a really useful tool when modeling neurodegeneration, because we can take these cells from patients of interest with specific mutations associated with Parkinson's, for example. And not only can we model the mutation present, but we can also capture their genetic background. So we can develop isogenic controls from these cells. And by doing this, we're really reducing variability due to genetic background in our experiments. So there has been extensive literature showing robust changes in neural activity in both animal and human models of Parkinson's disease, namely the increased firing throughout the basal ganglia, increased bursting activity, and synchrony throughout the nuclei of the basal ganglia. And then furthermore, it's also been shown that neural organoids can model certain aspects of neurodegeneration in terms of molecular pathology. However, there hasn't been any in-depth studies looking at if these network changes or changes in the electrophysiology that we see occur in vivo can also reliably be observed in organoids. So my question is, can we accurately model changes in network activity between mutant and control organoids? And so in order to do this, I need to first characterize basal activity in the organoids. So do we see reproducible patterns of activity, or is it more or less random? We can also characterize what are the main drivers of organoid activity. So what neuronal cell types play a role in the observed activity. And then we have to characterize activity in midbrain organoids that include these Parkinson's disease associated mutations. And so here we can look at how the activity differs over time compared to control organoids. So do we see robust changes in neural activity that can be used to describe a disease phenotype, and are they reliable and accurate? So to do this, I need to be able to record neural activity across the organoid so that I can characterize these patterns that are present in the neural network. And for this, I'll be using the Microelectrode Array, or the MEA. So MEAs consist of multiple electrodes embedded in the base of a well. Different MEAs can vary between having dozens to hundreds of electrodes. The ones I use in my project look something like this. So there are 16 electrodes organized in rows of 4, where each electrode is 350 micrometers apart. And so all electrodes are recording simultaneously, and what they record is the local field potential of the overlying neurons. So if you were to record and look at your raw data, you would see something like this, where you would have 16 boxes, so each box representing the recordings from a single electrode, and these vertical lines representing detected action potentials or local field potentials from the overlying neurons. Then we can also see the waveform of these action potentials, as well as a raster plot. So MEAs have a lower degree of spatial resolution compared to patch clamp EFIs, for example, but they can record from hundreds of channels simultaneously, and this makes them more suitable for measuring network activity or oscillations within your culture. And furthermore, they are non-destructive to your culture, so multiple recordings can be taken over long periods of time. So we can place the organoid over the electrode as such, and then once we're done recording, we get this output from the system that tells us the electrode that detected a spike, the time that that spike occurred, and the amplitude. And so from this raw data, we can look at different patterns of activity. And we do this primarily through bursts and network bursts. So bursts represent when a neuron fires very rapidly for a short period of time. They can do this repeatedly, and this tends to occur in more mature neurons. whereas network bursts represent when multiple neurons throughout a network produce bursts simultaneously. And so this signifies increased connectivity within your network. And so we can use this to extract a bunch of parameters that can give us an idea of what the pattern of neural activity looks like in our organoid. And we can also use this data to visualize network activity through a raster plot where we have the time of the recording on the x-axis, the spike train of each of the 16 electrodes on the y-axis, and then we can see that every little black tick represents a recorded spike. The highlighted blue boxes represent electrode bursts, and these vertical black lines represent periods when multiple electrodes burst simultaneously, so they would be network bursts. And so this is the general format that I'll be using throughout my, my talk when visualizing raster plots. So I currently generated organoids from 2 different cell lines. I have one cell line that's a synuclein triplication mutant. So here we have an extra copy of synuclein, which leads to Parkinson's pathology. And then it's isogenic control where that extra copy is knocked out. And so I generated about 200 midbrain organoids for each of these, which I let mature for 30 days before recording. This is just because in my experience, before being 30 days old, it's very rare to see any sort of spontaneous activity. And so once they reach this 1 month mark, then I start to record every 2 months for a total of 12 months. Currently, I'm at the 5 month mark, so there's still a little ways to go, but all the data that I'm going to show you today will be up until 5 months old. So after recording from the organoids at 1 month, 3 months, and 5 months, I can start to compare patterns of activity. So here in the orange, we see the AST23 mutant organoids, and in the blue, we see their controls. And at every time point, I record from 24 organoids in each group. And so the first thing that we can see is that these organoids really show minimal activity before 5 months of age. If we look at the total number of spikes that was counted throughout the recording, we see very little activity in the first month and the 3rd month, which increases at 5 months. And so this can be visualized through these raster plots again, where we see very random sparse activity in the first 2 time points. But at 5 months, this increases, and we even start to see the presence of electrode bursts and network bursts through our network. And so similar trends are seen when looking at other parameters like the mean firing rate, the total number of bursts present, the total number of network bursts, and the synchrony metrics, which is just a measure of how synchronous activity is across the network. And so when comparing activity between the mutant and control organoids, we don't see any differences in these early stages, but differences start to appear at the 5 month mark. So when looking at just overall activity, so such as this total spike count and the mean firing rate, these tend to be quite similar between the 2 groups, even at later time points. But where we do see differences is how in the connectivity of the network. So we see that the control organoids tend to show a lot more network bursts overall compared to the mutants. However, the total synchrony across the network is higher in the mutants compared to the control. So they seem to organize their activity differently. This I found was quite interesting when I first saw it, because I tend to think of these network bursts as periods of very high synchrony. So, I would think that if I see a lot of network bursts, that would mean that the synchrony across the network would be higher, but that's not what we see. Even though the mutants have less network bursts overall, they have a higher degree of synchrony. And so to look into this further, I tested parameters that could better describe the shape of bursts and network bursts and not just the overall activity to see if this is where the difference lies. And so to do this, I tested a few different parameters. So the average number of spikes present in a burst and the burst duration, then the average number of spikes present in a network burst and the network burst duration, and then the ISI within a burst. And so this is the interspike interval between 2 consecutive spikes within a burst. And so what we see is that the mutant organoids tend to have more spikes present within their bursts as well as a longer burst duration. So their bursts are stronger and longer, and similar trends are seen in the network bursts, where we have more spikes overall present in the network burst with a longer network burst duration. And then the mutants tend to have smaller interspike intervals within their burst. So this indicates that the spikes within a burst are firing much more rapidly in the mutants compared to the controls. And so this can all be visualized through raster plots like this. So we can see that in the AST23 mutants, although they have fewer network bursts overall, the network bursts are longer and stronger in terms of how many spikes they contain. So this can be seen through the spike histogram on top. And this is compared to the controls, which we can see have a lot more network bursts throughout the recording, but they're much weaker in terms of how many spikes are contained in them and their duration. And then we also see a higher proportion of overall spikes organized within the network bursts in the mutant organoids, with less random spikes outside of these network bursts. And this is most likely what contributes to the higher level of synchrony mentioned previously. So this can also be visualized by looking at the interspike interval distribution. So here we graph the ISI between two consecutive spikes on a log scale on the x-axis, and then the frequency of pairs of spikes that have that ISI on the y-axis. And so typically you would expect a bimodal distribution where you get one peak that corresponds to very small ISI times. And this represents the ISIs within bursts where you tend to have very rapid firing. And then you would get a secondary peak that corresponds to larger ISI times, which would indicate the spikes outside of bursts where you have much less firing. And so in the control, this is what we tend to see, where the first peak represents all the spikes that are organized in these network bursts and bursts. And then the second peak represents all these other spikes that are outside. And in the mutant, we see that almost all the spikes are organized in bursts, and therefore we only have this single peak of very small ISI times, since we have very few spikes outside of the burst and network bursts. So overall, the firing pattern seems to be a distinguishing feature of the AST23 mutant cell line at this time point of 5 months. And then finally, we can also run a principal component analysis on the 11 parameters looked at 5 months old. And this allows us to break down these variables into components that best describe our data set. And when we do this, we can see that we get 2 defined clusters that separate the mutant group from the control group. And this again is just to show that there's clear differences in activity between the 2 groups at this time point. So overall, we can see clear differences in neural activity between mutant and control organoids after 5 months of development, but not earlier. However, more recordings need to be done at later time points to see how this activity changes and evolves. And this needs to be done on multiple batches to test how reproducible these results are, as well as on different cell lines to test how robust these disease-specific phenotypes are. And then finally, we can also do a series of immunohistochemical analysis on all the time points to determine how the progression of molecular pathology correlates with observed changes in network activity. And so that's all to come still. So I'd just like to thank Dr. Fawn and Dr. Durkin for supervising my project, Meghna, V, and Paula for getting me trained on organoid generation, Carol for training me with iPSCs, and Relina for help on the EFIS analysis. And then finally, everybody at the EDDU and the Fawn Lab.
Okay, thank you very much, Ghislaine, for a great print, and thank you, V, as well, both of you, for great presentations. So our next speaker is Dr. Pierre Marquet, who's a professor in the Department of Psychiatry and Neurosciences at Laval University, and he's also the director of the International Joint Unit on Neurodevelopment and Child Psychiatry. And the title of his talk is Digital Holographic Microscopy, a high-speed label-free technique to resolve neuronal network activity. So thank you very much, Pierre, for being with us, and the floor is yours.
Okay, thank you for this introduction again and for this kind invitation. So just a few words concerning the research that we developed in the lab. We focus on major psychiatric disorder, especially major psychosis, including schizophrenia, bipolar disorder, and major depression disorder, which affect nearly 12% of the population. Unfortunately, in psychiatry, diagnoses are based only on clinical symptom and sign, which represent the advanced stage of the disease. So consequently, diagnoses are made late, and currently treatment are largely palliative. There is insufficient response with conventional pharmacological treatment and psychotherapeutic intervention. and also an evidence of illness progression and acceleration. Treatment targeting the period immediately preceding the onset of symptom called the prodromal period represents. There are many evidence that this prodromal period, when we can treat during this prodromal period, we have a more effective intervention. So finally, as for any disorder or disease, the sooner the treatment, the better outcome. Measured psychosis have also some have a neurodevelopmental component. This neurodevelopmental component has mainly been identified at the level of the neocortex, or more specifically at the level of the prefrontal cortex. There are many evidences. It's perhaps difficult to see what is written on this picture, but if I summarize, there are many observations suggesting that there is, for example, deficiency, myelinization, delayed maturation of inhibitory system, excessive excitatory pruning at specific period of the neurodevelopment. So a lot of different evidence suggesting the presence of this neurodevelopmental component during the childhood, but at the same time, we have no sign and symptom, specific sign and symptom during the expression or associated with the expression of this neuronal component. So it's a problem to detect or to perform a diagnosis because we don't have any clinical sign and symptom. It's a kind of paradoxical situation, because some people are speaking about measured psychosis as a brain, a kind of brain disorder of the childhood, but with no sign and symptoms. So the goal is to identify cellular risk biomarker with a specific or subtype of biomarker called endophenotype in relation with this neuronal development component. And at the same time, the goal, of course, to do that is to be able to develop earlier, safer, and more effective intervention, as just explained before, but also to improve the understanding of the pathophysiology or pathogenesis of this measured psychosis, because the pathogenesis is really completely elusive today. So what we have developed, it's a strong initiative to perform or to obtain iPS cells for performing a kind of modeling psychiatric disorder with these cells coming from offspring and patients derived iPS. I don't insist on this part because previous talk have already mentioned this kind of things, and I am not a lab which perform innovation in this part of research. We just use the state-of-the-art technique to perform this reprogrammation and neurodifferentiation. But what we can to do with that is to develop and to combine some new cells imaging technique to explore this different or this kind of iPS-derived cells culture. So to do that, it's important to be able to have access to high-risk cohort. High-risk cohort composed of the patient and the high-risk offspring deeply phenotyped from a clinical point of view. It's really important. To do that, we have installed an international joint research unit between L'Université Laval and Université de Lausanne, which have permitted to have access to two very quite unique cohorts, the Eastern Quebec Kindred study. It's a multi-general family study of people, of family affected by schizophrenia and mood disorder. We have approximately more than 1,000 family members with the relative and so on. And we have also access to the Lausanne Geneva high risk mood cohort with more than 300 offspring and 200 proband, which are also a quite long 20 years follow-up, which are very well characterizing. Again, I highlight this from a clinical point of view. So we have approximately reprogrammed 50 iPS lines from this high-risk cohort. Again, I don't insist on all these different steps, or programations and or differentiation, because it has been already explained before. But the goal is really to obtain cortical neuronal network, because as I mentioned before, this neurodevelopmental component has been quite well discharacterized, at least at the level of the cortical and the cortical part of the brain. So the goal is really to have this cortical neuronal network with aiming at identifying some phenotype and so on. But this culture have some characteristics which make them difficult to explore. They are highly heterogeneous cultures. The neurodifferentiation process take place over several weeks to a few months. And the characterization of this neuronal network, it's a challenge due to the heterogeneity. And there are different parts of the culture with different degree of advancement in the maturation and so on. So it's why we try to develop the imaging technique well suited to explore, to investigate these iPS-derived neuron cells culture and to identify specific functional phenotypes. It's a first step of our research to after that be able, when we have identified such a specific functional phenotype, to decipher in collaboration with other research groups, molecular mechanism and so on. So this kind of requirements that we have in such imaging technique to address this question of IPS exploration have to own some characteristic, to be non-invasive, to have a large field of view and high resolution approach, enable to capture many cellular features when we try to identify specific phenotype, and measurement should be performed on time scale ranging from milliseconds to several weeks, and also it should be compatible with implementation of automated approaches. So I will introduce now some, explain to you a little bit about quantitative phase digital holography and why we can do currently with this approach concerning the exploration of iPS cells. So a few words concerning the holography, which has been discovered by an Hungarian physician in the 40s, who had been awarded by the Nobel Prize during the 70s, Denis Gabor. I don't want to bother you with some physical concept, but it seems to me it's quite interesting, nevertheless, to understand just the key concept about or behind holography. So when we want to look at a specific specimen or macroscopic specimen, we illuminate the specimen with a light source, and after that, what we record on the camera, we record the scattered field, the interaction between the illumination wave with the specimen induce a scattered field, and the scattered field contain all the information concerning the specimens that we want to observe. Gabor proposed exactly the same thing, but to perform an interference between the scattered wave by the observed object with a reference wave. And so by doing this, we record an interference pattern. This interference pattern between the two waves, which is called the hologram. And after that, the next step is to extract the information from this hologram. Why doing this? Why we have to do this? Because we want really to record the phase, the state or the vibration of the light, if you want, the vibration of the wave. And this vibration in the visible range is very fast. It's approximately 15 power 15. So it's impossible for any sensitive media to record this state of vibration. But by performing this interference, the phase is coded in this interference pattern. So the next step is how to extract the information, which is coded in the interference pattern. It's Gabor proposed to re-illuminate the photographic plate. By doing this, he recreates an exact replica of the scattered wave, of the wave scattered by the object. So if you look at this photographic plate re-illuminate by a reference beam or by just a light source, you will see, or you will get in your eyes this scattered wave, the exact replica of the scattered wave by the object. So you will see the object, because there are no difference if you look at the scattered wave when the object is present or when the object is no longer present, because it's an exact replica. That's a key concept. But the important point is that we can record the whole things concerning the scatter wave, the amplitude and the phase. And what the phase is important, I am sure that you know very well what it's called in biology, the phase contrast microscopy. It's a bright field image of cells in culture. Here you can see some you can see two cells because they are pigmented cells. It means that they absorb the light. But when you perform a classical phase contrast image, you can appreciate a lot of things, a lot of details concerning these cells. Why? Because a phase, as illustrated on this scheme and at the left of the slide, has, at least from an optical point of view, a refractive index which is different from the surrounding medium, and thus it provokes a phase delay. So with a phase contrast, we can measure something in relation with this phase delay. So this is the root of the contrast. So how we do that in practice? We just have built a transmission microscope, and we perform this interference, as I explained before, to record the phase of the scattered wave by the object. So in practice, we have developed this kind of holography technique in the context of digital holography microscopy. It means that we use a CCD camera to record the hologram. Here we use, of course, a microscope objective, and we have our reference wave. And the key point is the fact that we don't need to reilluminate the hologram to extract the image. We can perform a numerical reconstruction, because when we know the phase and the amplitude of the scattered wave by the observed specimen, we know very well how to numerically propagate the waveform. So we do that numerically. And here, for example, it's an image of the USF test target, the classical test target. When we perform the classical amplitude or intensity image of this phase target from the hologram reconstruction, we use this conventional image, which is diffracted limited, just to illustrate that we have a good lateral resolution. But when we reconstruct the quantitative phase image, it's the same of the same object. You can appreciate we get a lot more information concerning the observed specimen. And just a few points concerning the advantage of using a numerical approach to do this reconstruction is the fact that we record a single hologram. The reconstruction rate can be performed at more than 25 seconds with a classical personal computer today. And during the numerical reconstruction, we can correct from any aberration introduced by the microscope objective. So we can use very cheap microscope objective. But what is more interesting for us is the fact that we can reconstruct the image, because it's a numerical reconstruction, at different distance. So it means that we can obtain an extended depth of focus images. It means that if we want to observe during a long time some different process in relation with the node differentiation, we can permanently adjust, not during the acquisition, but after that, as an offline treatment, some defocalization and so on, just from a single recorded hologram. And another interesting point is that we have a nanometric axial sensitivity, because it's an interferometric technique, and by such technique, it's possible to monitor the phase with an extreme sensitivity. Just to give you an example, here it's red blood cells. Red blood cells are known to exhibit a flickering of their membrane. It's an oscillation of their membrane at the nanoscale. We can easily appreciate this oscillation of the red blood cells with this kind of techniques. So what we do more concretely, we have our cells culture in a perfusion chamber, and what we measure, it's this delay. I speak about this delay, the phase retardation. In fact, we measure the phase retardation between an area where there is no biological material, it's Phi2, and where the light has passing through the cell, so it's Phi1. And this phase delay is proportional to the cell thickness, not a big surprise, but the root of the contrast is really the difference between the intracellular refractive index and the extracellular refractive index, which corresponds to the perfusion solution. So this is a typical image that we obtain. It's a cortical neuron. It's not an IPS, but it's a mouse cortical neuron. And the important point that each pixel represents this phase delay that I have just explained before. And we have also this to illustrate the sensitivity, the high sensitivity that we have, which is approximately a sensitivity corresponding to a 10 nanometers fluctuations. So now what we can do with this quantitative phase signal in terms of biological process, that's the next step that I want to explain. So the first observation that we did is the fact that this phase signal is highly sensitive to water movement. It's, for example, an osmotic shock, a very simple osmotic shock that we perform on neuron in culture. Each curve represents the central part of the neuronal body, and we have the time course of the phase. When we perform this hypotonic shock, we have a huge phase decrease. This phase decrease reflects the fact that we have a dilution of the intracellular refractive index. So we have a decrease of the intracellular refractive index, which become closer to the refractive index of the extracellular medium. And this sensitivity or the sensitivity to the intracellular refractive index, it's much stronger than the sensitivity to the modification of the volume, because we have also an increase of the thickness with such a hypotonic shock. But what we measure, that's really the dilution of the intracellular refractive index due to the transmembrane water movement. So the next step was to combine the DHM with an electrophysiological approach, because when we activate a neuron, here you have a quantitative phase image when we perform glutamate on a neuron, and we see a strong phase signal. This means that probably there are some transmembrane water movement associated with the electrical activity of the neuron. So the next step was to combine to what we can deduce one signal from another. So to establish the relationship between the phase signal and the transmembrane current, we performed some experiment on X cells which express GABA receptor. I don't want to go in details, but directly to the main message, what we did, we clamp at different value the membrane potential of these X cells. We applied GABA, and we record at the same time the transmembrane current and the phase response. And what we see, for example, when we clamp at minus 100 millivolt, we know that we have an exit of negative ion, the chloride ion, which corresponds to an increase of the phase signal, which means that we have an exit of water. When we do the same things for when we clamp the membrane potential at 40 millivolts, we have another situation. Here we have an entry of negative ion, the chloride ion, and we have a decrease of the phase signal, which corresponds to an entry of water. So here's the main message. Each time that we have an ion movement through the membrane, we have water fluxes associated for osmotic reasons. So we can measure with CTHM this transmembrane water movement. So we can follow by monitoring this transmembrane water movement, the ionic activity, if you want. So to illustrate, another way to illustrate that, it's we perform a classical IV curve that with electrophysiology. So the relationship between the current and the membrane potential and the current to determine the equilibrium membrane potential, mainly for the chloride. And we can do the same by monitoring the amplitude of the phase response, and we can detect purely optically the equilibrium membrane potential for the chloride here. So the next step was how to find a relationship between our optic signal and the transmembrane water current. So to do that, we use very simple relationship, a linear relationship between the intracellular refractive index and the concentration of the different components in the cell, and the fact that the water the variation of the volume is proportional to the number of transported charge. So it's mean an integration, temporal integration of the current, net charge through the membrane. And we use this relationship that I explained before. By doing this, we were able to establish a relationship between the current and the phase. So it could, at the first glance, it could seem complicated, but it's not, because we have not a lot of parameters. We have the phase that we can measure, and we have a temporal derivation. We have the volume at the beginning of the stimulation. And we have two parameters. One, R, correspond to how the volume is changing, how the cells changing his volume. We can also measure this part with DHM. And another parameter, that parameter, we postulate, it was an obligation to add such a parameter to have a coherence with the unit. So we know also the unit of this parameter, which corresponds in fact to the number of water molecule associated with the net transported charge. So the only parameter that we have to find to establish the relationship is the epsilon. So again, it's again the stimulation, the neuronal stimulation with glutamates, which are presented on this movie. And here you have the two relationships, the two curves, the transmembrane current and the corresponding phase response. You can appreciate the temporal derivation between these two curves. And by applied the relationship, the equations that we have established before, we were able to compare the predict current with the real measure current. And to do that, we have to fit to determine the epsilon. And the epsilon, when we perform by a least square algorithm, this adaptation, this fit, we find that epsilon is quite huge value. It means that's the main message, that there are a lot of water associated with the net transported charge during a neuronal activation. It was quite surprised at first, but the evidence are stressing this point. So there are also a refractive index associated with this neuronal activity, which is quite high, but it's probably not a physiological condition. So the next step was to have a better understanding of this phase signal. To do that, we combine DHM with fluorescence microscopy. Again, I don't want to go in details with that. But what we obtain when we do that, we have at that time the phase signal when we again perfuse glutamate on neurons. We have this phase response in black, and we at the same time we monitor calcium and sodium. And one we can appreciate by doing this, that we have the first step, which corresponds to the opening of the AMPA-K-NAT and MDR receptor with a huge SADIC current. And after that, we have a recovery. And by performing some pharmacological experiment, it was possible to determine what is the key element dictated this recovery. There are mainly two elements, the NKCC1 and the KCC2. And the NKCC1 is act in a prolongation of the depolarization, and after that, the KCC2 is responsible for the recovery. So this is the main element implicated in this response. After that, we don't know exactly the role of aquaporin because it's not very well known how water goes through the membrane. So glutamate firing again induces a significant water flux. So now you have understand that it's possible to follow the neuronal activity, the neuronal maturation by following this quantitative phase signal. And by doing this over a large number of cells in a culture, we found mainly three types of responses: a biphasic phase response, a reversible phase response, and an irreversible phase response. So irreversible phase response is associated with an excitatory effect, which is well known when we perfuse glutamate on a neuronal cells culture. So this is an irreversible response because we have a subsequent cellular death, which has been demonstrated by this article when we can just predict the subsequent neuronal death by analysis the slope, because it's a very drastic slope at the beginning. And we can also protect the neuron. It's different experience we will perform by adding lactate on this culture to convert an irreversible response in a reversible response. Now, the interesting part is why we have these two kinds of response, the biphasic and the reversible response. By analyzing a little bit more accurately these two responses, in fact, it's really due to an interplay because NKCC1 and KCC2, when we have the biphasic, it's a strong activity of KCC2, and we can relate this strong activity to NKCC2 with a strong connection with the inhibitory system on a connection on the specific neurons and we analyze. So when we have strong, because what I have to explain before, when we activate a neuronal network with glutamate, we activate not only the excitatory neuron, but we also activate the whole network, so the inhibitory neuron. It means that when we follow the response of a specific neuron, we have the integration of the excitatory connection and the excitatory input and the inhibitory input. And we see that when we have this different kind of response, it's the details of the mechanism. In fact, we activate with glutamate the excitatory connection through glutamate and the inhibitory connection through GABA. And the chloride activated in a different direction, the opposite different direction in which chloride activate KCC2 and KCC1, and the calcium and sodium activate in the opposite direction, again, KCC2 and KCC1. So when we have a specific system which predominates, we have a biphasic response, and when this other which predominate, we have a reversible response. So it's interesting to see that the analysis of this shape of this response can inform us not only on the maturation, on the neuronal maturation and neuronal activity, but also on the close connectivity of this neuron with an inhibitory input system and an excitatory input system. And we perform the analysis of this different response during the neuronal maturation of the neuronal culture, and we see indeed that we have at first rather a few biphasic response, and after that we have an increase of the biphasic response and a decrease of the reversible response. We reflect the maturation of the inhibitory system. And by the way, we can also monitor what we call the GABA switch, but I don't have time to present these kind of things today, and I have to conclude. So I really try to convince you that with the analysis of the quantitative phase signal, which is a non-invasive, label-free signal, we can have a lot of information concerning the maturation of the neuronal culture, the local connectivity, structural connectivity, and local connectivity. We have also a lot of technical improvement to achieve. I just mentioned some of them, which are quite interesting because I just a few words concerning the signal to noise ratio, because during all my presentation, I speak about the neuronal body, but I really want also to address the visualization of the connection. And I will show you how we can do that. And also some interesting points, sorry for that we can modulate. We can extract a lot of different biophysical parameters from this quantitative phase signal. I don't have time to go in details, but absolute cells volume, water permeability, transmembrane water movement, I explained to you, biomechanical properties, cell membrane fluctuation. But we have also trying to visualize inside the cells by modifying the extracellular refractive index. So here we have new developments that we perform to decrease the signal-to-noise ratio and to have a clear visualization of the neuronal process to address the same things that I show you, but at the level of the neuronal process, dendrite, and so on. And by modulating the extracellular refractive index, we can completely change the information that we get with this quantitative phase image. By doing this, we have really access, for example, to a visualization of the nucleus, the intracellular nucleus. It's a preliminary data. We have a low magnification, but we will continue this aspect to really have a vision of the intracellular compartmentation of this kind of cells. So it's very complement to, this is enrich the information that we can get, and it's always label free. So thank you for your attention. And I want to thank all the guy in my lab involved in this neurotechnological development imaging. Thank you very much.
Great. Thank you very much, Pierre. Thank you, V and Ghislaine as well. So just to recap, we had one grad student, one postdoc, and one PI, and all three took the entire time for their presentation, not leaving any time for questions. So look, normally it's coffee break right now, but we'll take maybe 5 minutes, 10 minutes. I think the next session is in how much time, Mario?
Yeah, we still have time. We have the coffee break until the 2:45, so that means we're good. We're good. We can take questions.
All right then. So maybe we can start by sections if there are questions about V's presentation, and then Ghislaine, and then Pierre. If we can, I'd rather let the trainees ask their questions first, if there are any trainees in the audience, and then we can get the PIs as well. But otherwise, then please feel free to either type your questions or just, we won't be able to see raised hands. So you can maybe just unmute yourself and ask your questions.
Hello.
Hi, Vincent.
Yeah, okay, I have a question for the second talk, actually. And I'm not a trainee, so I don't know if I can go because I'm breaking all the rules.
It's all right. It's all right. No, go ahead.
Okay. I was wondering if there's any, if you have looked at the spatial components of your network activity. So do you see a very homogeneous response of the inside the entire org? So it's kind of shifted more toward, I guess, the upper layer of the organoids. And how do you actually control from that when you record from a wild type or like non-mutant population and a mutant population?
Yeah, so for the organoids, the mature neurons tend to be on the outer layers, and the center of the organoids tend to be a necrotic core, so there's not a lot of activity in there. One limit of the MEA, especially the 2D MEAs that I use, is that you can only get one side of the organoid and only get, the outer layers, the activity from the outer layers. So that's something that I'm working with other MEA companies and demoing some of their products to see if we can get a better image of the full, the full scope of the organoid. Currently, this isn't controlled between the mutants and the controls. They just put on the MEA well, and whatever side is on gets attached to the laminin coating, and that's the side we record from.
I see. Okay. And so you're using a 4x4 MEA or a 16x16? I don't remember.
4x4, yeah. So 16 total, yeah.
So you don't see any difference between in terms of the spatial distribution of your activity onto these these 16 channels? Not so much.
No, not so much, yeah.
Thank you.
I have a question for both the first two presenters. I was wondering, in these organoids that you generate, have you tried introducing perhaps a GCamp at the early phases when maybe before reprogramming the cells, so that once you have the full organoids, you would be able to follow network activity just by imaging and see if there are any waves of excitation that you can follow instead of just the MEA, because like you said, it's just one side from which you can record, whereas being able to use the GCAN signal, you may be able to see the entire activity of the network.
So I don't know about V. I've tried to do some calcium imaging on the organoids, but we haven't, like this was at much later stages and I wasn't very lucky, wasn't able to see a lot of activity. I haven't tried integrating a GCAMP during development, so that would be interesting to try.
I think it's an excellent suggestion. It should work very well. We are always limited by the 3D structure itself for the imaging. So yeah, this is something to try and to optimize because the tissue is quite deep. So yeah, it could be challenging as well.
Maybe this is Ted Vaughn. I can just add that with Yves and Marie-Ève, we have just received a grant to do exactly that kind of work. So it's in the plans.
Very good. Yeah, thank you.
I have a question about the necrotic cores of the organoids. Are there ways to like supply oxygen or nutrients to these neurons so that they don't die?
Yes, so the first protocol that we published in 2017, 18, I used to do them in the 96 well plate, and for that reason, they reached 4 millimeters in diameter. So it was huge, and of course, then the penetrance of the oxygen and nutrient was quite difficult. So there are plenty labs around worldwide who are trying to implement, tubings inside to provide a better diffusion first of the growth factor to control better the differentiation of the tissue, but also like to provide more nutrients within the center. And the second methodology that I published, and that was what I presented today. So the idea with this EBEDs was to really constrain them in space. And so instead of growing up to 4 millimeters, they are actually just 1 millimeters. And that does help a lot for of course, the nutrient penetration. Also, we switched the system. Instead of having them on the 6-well plate shaking on orbital shaker, I use now a spinning flash bioreactor. And that also does help for the proper diffusion of the nutrient and oxygen. So actually, they are much more better than the initial protocol.
Okay, so you'd rather have smaller but healthier organoids, quite simply. Okay.
I've run data to compare, like, my triplication organoid versus my isogenic control one with both protocol. And I did observe the same pathological features in both. So I was able to recapitulate all the main features in both systems.
Okay, thank you.
I have one more question to V this time about the staining that you did on your organoids, using the MAP2 antibody. It's striking that a lot of the signal seems to be peripheral, and not at the center of the organoids. I was wondering if you had an explanation for that.
So for the cryosection, it's quite everywhere where there are the neurons, but for the 3D stainings, so the clearing one, the video I showed you in 3D, of course, again, because this is a 3D structure, you have to manage the penetration of the antibodies. And Maria, depending on the antibodies, she has to incubate for several days with primary and secondary antibody. So there's the fact that the cells organize in space. So for sure, you will have like some sort of area enriched in the premenergic neurons or neurons or astrocytes. And you have the other technical components with the penetration of the antibody that can make it seems like the cells are much more stained on the periphery. So I think there are both factors here.
Okay, so it's just an accessibility issue.
Exactly, exactly.
Thank you.
I have a question, if I may, for Ghislaine. So you showed that your recording, the activity increased drastically at 5 months, not that much activity before that. So I'm just wondering if you try to wait longer and what you think you would get.
Yeah, so my project is still ongoing. So I do plan to go up until 12 months to record activity up until a year old. What I've seen in some preliminary data that I used when I first started in the lab, these were on about 200-day-old organoids, and I saw that we still see these very strong network bursting activity, but they tend to cluster into groups. So you get these periods of clusters of network bursts, and then silence, and then another cluster, and there tends to be their firing pattern. We have yet to see if we'll see the same thing in these organoids. It was the ones I had seen in my preliminary data were also synuclein triplication mutants, but they were a different cell line. So it'll be interesting to see if once I reach that stage in development, if we see similar patterns of activity.
Great. Thank you. So, Paul, did you have a question?
Well, I was just following up on this increase in activity after 5 months. Does this reflect more synapses? Can you tell, or does this reflect, I don't know, disappearance of some inhibitory neurons in the network?
So I think more synapses for sure. So increased connectivity within your network. And I think what I've seen in the literature too is that around, if I'm correct, 3 months, we start to get more oligodendrocytes and astrocytes, and this really helps with the activity. So they're very underdeveloped, I would say, before that age, and so we would expect them to be more silent.
Thank you.
Okay, so if there are no more questions, maybe we can go on to take that coffee break, and make sure, please, that you're back here at 2:45. So that is in 17 minutes for the next session.
I guess now we have 2 former students, but now have become independent scientists, Canadian icons, you name it. We have Stephanie Borglen and Jean-Claude Beek. So Stephanie from Hodgkin Brain Institute in Calgary, as you can tell from this beautiful picture. She's going to tell us about how they dissect optogenetically the circuits involved in reward seeking. And then Jean-Claude from Ottawa will tell us how they integrate computational, electrophysiological, and optical approaches to study synaptic and network dynamics. So let's fire up and start with Stephanie. Please be ready to answer everyone else, everyone else questions in the Q&A section, and we'll try to cover them as we can. So Stephanie.
Great, thank you so much. And thank you for inviting me. It's a real honor to be on this panel with all these other outstanding speakers doing just mind-blowing science. So it's really nice to be here. Okay, so what's going on with my screen? Okay, dopamine neurons. So most people know that VTA, or mesolimbic dopamine neurons, are kind of a hub of activity that respond to salient events that really guide motivated behavior. So VTA dopamine neurons are really involved in that cost-benefit analysis to decide if you're going to put in effort to obtain a reward. But one thing that's lesser known is that, in fact, this population of dopamine neurons is actually quite heterogeneous. So these neurons have unique electrophysiological properties, and they also have independent inputs and projections to different regions. And and these neurons also respond differently to drugs of abuse or rewarding or even aversive stimuli and stuff. So this is just sort of a diagram that I'm not going to take you through because it's incredibly busy, but one of the things that really governs the activity of these dopamine neurons is that they receive different afferents from a variety of different excitatory or inhibitory inputs from brain regions, and they have different output projections. And of course, dopamine in the nucleus accumbens may be involved in engaging this effort in obtaining rewards, but they play a completely different role in the medial prefrontal cortex. So obviously the effects of dopamine are really guided by their the inputs that project to dopamine neurons, and of course their consequent outputs. And so my lab's been somewhat interested in characterizing some of these inputs, in particular from the lateral hypothalamus. So orexin neurons are produced solely within the lateral hypothalamic and the parafornical area of the hypothalamus. And they have projections of what pretty much widespread throughout the brain and stuff, including a projection to the ventral tegmental area. So the function of these orexin neurons are basically to help transform motivational action into adaptive behavior. And so basically these neurons are most active when the animal is engaged in reward seeking, or when the animal is stressed or escaping a stressor, or when there's a highly motivational event like a sexual opportunity or that type of thing. And of course, these all occur during wakefulness periods, and these neurons are quite quiescent during sleep periods. So basically, these orexin neurons serve to sort of mark salient events, and dependent on where their projection targets are, they guide motivated action. So the odd thing about orexin neurons is that it's co-localized with another peptide called dynorphin, which is an opioid peptide. So while orexin is primarily excitatory, it acts on orexin 1 and orexin 2 receptors, which are primarily GQ coupled receptors. And typically orexin will increase firing and that type of thing. Whereas dynorphin is primarily an inhibitory peptide. So it acts on kappa opioid receptors, which are typically coupled to GIGO, which usually lead to a hyperpolarization of cells or a reduce in release probability or that type of thing. So why these two peptides are co-expressed and later found to be even packaged into the same dense core vesicles and stuff, suggesting that they're potentially released at the same, in the same place at the same time. So that was one thing, that was one question that we were really interested in, is why do you have this excitatory peptide and this typically inhibitory peptide being packaged in the same vesicle and co-released at the same time, or are they co-released? And so, we collaborated on this project with the Mouchant lab, and one of the things that we found is that when these peptides were co-applied together, they sort of null out each other's effects. So orexin increases firing rate and dynorphin decreases firing rate in the ventral tegmental area. They, when applied together, the firing rate was unchanged. However, there's a lot of variability in this, and I'm going to discuss that on the next slide. But the other thing that they found is that dynorphin may be acting like a brake on reward seeking with orexin. So, for example, when orexin is released, it promotes reward seeking. However, if dynorphin is co-released, you have this brake on reward seeking. So you can sort of enhance this brake by blocking orexin receptor activity, and that's why hypocretin receptor antagonists seem to be quite effective at reducing, reinstatement to drug seeking and that type of thing. So, as I mentioned before, we were really interested in how these neurons influence dopaminergic activity and influence reward seeking. And so we really wanted to sort of test this idea is if you in these experiments, they were primarily done with exogenous application of agonists and antagonists and stuff. And we were curious whether or not you know, if you drive this neuronal population and stuff, could you get co-release of these two peptides in the VTA? And so what we did is we used orexin Cree mice, and we administered a virus containing channel rhodopsin or a control virus containing mCherry. And then we implanted a laser in the ventral tegmental area, and we stimulated terminals of these LH orexin or dynorphin neurons within the VTA. So this just is an electrophysiological slide just showing basic characterization of channel rhodopsin within the orexin neurons, showing that indeed they can follow, you know, 5 hertz, 10 hertz, 20 hertz, and up to 30 hertz and stuff quite reliably. So for our experiments, we're primarily stimulating in the behavioral experiments inputs at 20 hertz, which is fairly typical in inducing these behaviors. So the other thing, so we did look at what, you know, so right now we're recording from a VTA dopamine neuron, and we did check to see if there was, you know, a difference in 5 hertz versus 20 versus 30 hertz. And we, 5 hertz did not induce much change in the firing activity of dopamine neurons. However, 20 and 30 hertz in general increased dopamine neurons. But I'm going to talk to you a little bit later on why this is so variable. So, so the first question was, does optogenetic stimulation of the LH orexin dynorphin input promote place preference? And so in these experiments, we are optogenetically stimulating lateral hypothalamic inputs to the VTA. And we used a real-time place preference assay where the animals or the mice were habituated in the absence of stimulation to the 2 compartments. And then during the training session, the animals received 3 one-hour-long training sessions where the stimulation occurred in one specific compartment, and this was counterbalanced. And then on the post-stimulation session, which was a day later, we tested which side the animals preferred. So basically, in the center one here during the real-time place preference sessions, the animal can choose either no stimulation or choose stimulation of the orexin inputs in the VTA. And this basically asks, you know, does the animal prefer this stimulation of these inputs? And then the last session asks, does the animal prefer this or remember this stimulation? So this example here is in the control virus, and you can see with the control virus, there's no difference in this stimulation zone versus the no stimulation zone. However, in the presence of channel rhodopsin, you see a strong increase in preference for the stimulation zone, and this increases over time. So in the 3rd session, where they've received the most pairings of the stimulation, they have a strong preference for the stimulation of the lateral hypothalamic orexin dynorphin inputs compared to these mCherry mice with the null virus. And then finally, in the post-stimulation session, so they're not actually receiving any optogenetic stimulation of these inputs, they continue to remember their preferred side. The critical thing, like obviously orexin neurons are involved in arousal. And so one of the things we're concerned about is that does driving this input into VTA dopamine neurons alter locomotor activity. And so we measured distance traveled and velocity, but we did not see an effect of channel rhodopsin or stimulation of these inputs as there was no difference between the mCherry controls and also no difference in the stimulation off compartment versus stimulation on compartment. So suggesting it doesn't alter locomotor activity. So the other question is that does this in fact require orexin 1 signaling? So presumably, you know, when you're stimulating these inputs, you're causing release of whatever the fast transmitter is, and typically in these neurons it's glutamate, but also orexin and dynorphin. And we found that we were able to replicate this effect, this increase in preference over time in the presence of a vehicle. However, in the presence of the orexin 1 receptor antagonist, this SB compound, there was an inhibition of place preference. So suggesting that, in fact, orexin is mediating these effects by acting at orexin 1 receptors in the VTA. So we also tested for effects of the antagonist on locomotor activity. And even though there was no difference in locomotor activity between the on compartment and the laser off compartment, we did see a main effect of the drug on distance traveled and velocity. However, even though this drug does influence locomotor activity slightly, there was actually no effect on preference, because it equally reduced locomotor activity in either the stimulation off compartment and stimulation on compartment. So the next question is, what is the contribution of dynorphin to this? So again, we replicated our effect, whereas stimulation of these inputs causes a preference to the context associated with that stimulation. However, in the presence of the kappa opioid receptor antagonist, NorBNI, there was still a place preference. Interestingly, there was no longer significance in the post-stimulation session. So in the absence of stimulation, these animals no longer recalled that prior preference in the presence of the kappa opioid receptor antagonist. Again, we looked to see if there were any differences in locomotor activity, and NorBNI did not alter locomotor activity between the on and off compartment, although there appeared to be a slight increase in velocity with NorBNI as a main effect. But again, no difference between stimulation compartments. So the next question is, are these effects mediated by dopamine? If you stimulate this particular input, does it actually increase dopamine in the nucleus accumbens? And so to do this, we used a fast scan cyclic voltammetry, which measures sub-second dopamine concentration. And so we used an electrical stimulator in the VTA to evoke dopamine concentration. And we also used a blue laser to stimulate the lateral hypothalamic inputs to the VTA at the same time. And basically we're asking, is stimulation of these terminals in the VTA modulating evoked dopamine? So this is just showing you electrical stimulation on its own, and we're able to, using 60 hertz, 60 pulses, we're able to do electrical, we are able to electrically evoke dopamine. Interestingly, on its own, laser stimulation of the terminals does not cause dopamine release. So stimulating the orexin dynorphin input to the VTA by itself does not increase dopamine release. However, what we found with electrical stimulation plus laser stimulation, you actually get a potentiation in the response. So comparing the null virus to the channelrhodopsin, you see an increase in, in dopamine in the channelrhodopsin, the electrically evoked dopamine release with the channelrhodopsin. And so this is measuring peak dopamine, where you see an increase, and area under the curve, where you also see a significant increase. So suggesting that orexin or orexin dynorphin is playing a modulatory role in potentiating dopamine release. So this is testing whether orexin receptors or dynorphin receptors are involved. And so in the M-cherry mice, which are the control, we see no effect of the agonist, we see no potentiation of the response. In the channelrhodopsin, the pre-SB which is the green line here, is potentiated compared to the controls. However, in the presence of the antagonist, you see a return to electrical stimulation dopamine levels. So suggesting that you can evoke dopamine release, and after or you can potentiate evoke dopamine release by stimulating these inputs, and this is blocked by an orexin 1 receptor antagonist. So looking at the kappa opioid receptor involvement in this, and in the pre-NORBNI, we see a potentiation of a laser evoked potentiation of electrical dopamine release. However, NORBNI has no effect in this. So suggesting that kappa opioid receptors are not regulating the increase in dopamine release, but orexin receptors are. So in the third experiment here, what we've done is we are recording an NMDA current. So one of the questions is, how could this potentiation of dopaminergic activity be working if you're through orexin 1 mediated receptors and that kind of thing. And we thought, well, one of the things is that orexin could be acting at other inputs to promote, glutamate release and strengthen these synapses and stuff, and allowing for increased or easier burst firing or that type of thing. So we recorded evoked NMDA receptors for this. And we found that when electrically evoked NMDA currents and stuff, we could potentiate them by stimulating the lateral hypothalamic orexin dynorphin inputs. And we saw a fairly consistent potentiation, although it wasn't particularly huge, with laser stimulation. We looked to see whether or not this effect required orexin 1 receptors. And when we did this experiment in the presence of the orexin 1 antagonist, in fact, we saw a slight depression of the response. So this suggests that something else might be going on here and stuff like that, that, in the absence of orexin signaling, you have something dominating. Maybe kappa opioid receptors could be involved in this. So we were next curious about whether or not how this photostimulation influences the firing of dopaminergic neurons. So this is just showing in the control virus that the light stimulation itself does not influence firing activity in the VTA. However, what we noticed is that when we stimulated the lateral hypothalamic orexin dynorphin inputs, we sometimes got a potentiation, and we sometimes got no effect, and we sometimes got an inhibition of firing. And so we've kind of quantified this, and mostly we get inhibitions of firing, and sometimes we get an increase in firing. So we were wondering, could that be due to either orexin? Could the potentiations be due to orexin signaling, and could the depressions be due to kappa opioid signaling? And so if we saw an increase in firing, we then applied the orexin 1 receptor antagonist, and typically that brought it back to baseline. Similarly, if we saw depression, we would apply the kappa opioid receptor antagonist, and this again brought it back to baseline. So this is sort of preliminary studies suggesting that the increases might be due to orexin signaling, and the decreases might be due to kappa opioid signaling. So we were wondering, is this firing induced by synaptic transmission? Are the reasons why we're getting increases and decreases in firing is not necessarily due to peptidergic content, but is it due to, we're just releasing fast transmitter and stuff, and that's influencing postsynaptic responses. And so we use synaptic blockers to prevent signaling from at either excitatory or inhibitory receptors. Fast transmitter receptors. So even in the presence of, the synaptic blockers, we still saw an increase in firing and stuff. And this increase in firing was blocked by SB, the orexin-1 receptor antagonist. And similarly, the decreases in firing were blocked by NORBNI, and this is all in the presence of synaptic blockers. So suggesting the peptides themselves are actually influencing we're optogenetically releasing peptides themselves, and this is influencing the firing rate of VTA dopamine neurons. And so in this experiment, we are blocking both the effects of the receptors. And when we pre-apply both the orexin receptor and the kappa opioid receptor antagonist, we prevent any change in firing rate. So then we were wondering, is the reason why we get sometimes increases and sometimes decreases is because, we're actually recording VTA dopamine neurons that project to different regions, and they have a different, opioid or orexin receptor complement. And so what we did is we used retrograde labeling from either the lateral accumbens shell or the medial accumbens shell or the basolateral amygdala. And we recorded those neurons with known projections while stimulating the lateral hypothalamic input to those VTA dopamine neurons. So in these cells, which we're looking to the lateral accumbens shell projections, we find that we can induce an increase in firing that appears to be orexin dependent most of the time. So about 65% of the time, whereas a decrease in firing occurs, 36% of the time. So suggesting, that in this projection, orexin kind of predominates in modulating it. In the medial accumbens shell, in this projection, although we have a few cells that cause a very large increase, for the most part, or 63% of the time, we see decreases. And these appear to be blocked by NORBNI. And in the basolateral amygdala, out of the cells we recorded, 100% of them were decreased responses. So dopamine neurons that project to the basolateral amygdala are primarily modulated by dynorphin from the lateral hypothalamic input. So to summarize this data, we see, when we stimulate the lateral hypothalamic orexin and dynorphin inputs to the VTA, we see an increase in place preference that is acting through the orexin-1 receptor signaling. We see increases in evoked dopamine in the nucleus accumbens, and this is also acting by orexin receptor signaling in the VTA. And we see that this potentiates NMDA currents of dopamine neurons. We also showed that modulation of VTA dopamine inputs from this orexin dynorphin input is projection dependent. So you get bidirectional modulation of firing in the lateral accumbens shell, which is orexin dominates over dynorphin, and in the medial accumbens shell, dynorphin dominates over orexin. And finally, decreases in firing the BLA projections that is primarily dominated by the dynorphin release from this particular input. So this is just sort of a summary figure. So I'll pop back to that in case you have any questions. But before that, I just want to thank the people that did the work in my lab. So Ida Mohammed Khani did all of the electrophysiological recordings and retrograde labeling and that type of thing. Catherine Thomas did all the fast scan cyclic voltammetry and the RTPP behavior. Medea Rana was a talented undergraduate student that helped Catherine out on a lot of these experiments. And Corey Bamo, who's moved on to his postdoc and is now looking actually for a faculty position in Canada, and he's terrific. And he sort of had the original conception of this project by applying exogenous agonists in these known protection targets to understand how they work. And so I'd be happy to take any questions.
Great, thank you, Stephanie. That was great. We have about a couple of minutes before the next presentation, so we can entertain some questions now and then continue maybe at the end if there's more time. And actually, there is one in the chat from Rogerio Francavilla, and he's asking, do you think orexin, as other peptides, needs high frequency stimulation, higher than 30 hertz, to exert its effect and to observe a stronger difference in mouse behavior. And he has a follow-up sort of question saying, any insights from in vitro electrical stimulation studies involving this, focusing on this circuit? Thank you.
Yeah, that's a good question. I don't know about higher than 30 hertz and stuff. We haven't actually measured whether or not the lateral hypothalamic neurons can follow frequencies faster than 30 hertz and stuff, but typically higher than 30 hertz is quite high, like at least driving the soma for that, like perhaps driving the terminals. You can induce different like LTP and LTD using like high frequency or lower frequency stimulations at much greater than 30 hertz. But it would be an interesting question to test. Although we did see, most of our behavior studies were done at 20 hertz, and we did see quite strong real-time place preference to observe these effects, and it seemed to be mediated by the peptide. One thing that I've been asked before, and I don't necessarily have the answer to completely, is it possible that the reason why orexin predominates during these effects is because we're using a stimulation paradigm that is actually evoking orexin release and not dynorphin release, and maybe dynorphin has different stimulation requirements, release requirements, and notably, those are packaged in the same dense core vesicles and stuff. So presumably the release requirements would be similar. I think what the net effect is going to be is basically where you're stimulating and what receptor complement is expressed postsynaptically. But, and also I think I've seen several papers where they're electrically evoking opioid release with much lower frequencies, like even a paired pulse has been shown to evoke opioid receptors in regions where there's super high expression of endogenous opioids, like the intercalated neurons of the basolateral amygdala. So yeah, I think, the release requirements obviously depend on the brain region where you have that expression, whether you're looking at somatic dendritic release or terminal release, but yeah, we haven't tried higher than 30 hertz to answer the question. Sorry about that. Making a long answer.
No problem. We have to move on to the next. I'll ask you another question, but maybe you can unshare your screen now.
Oh, yeah.
And Jean-Claude can start sharing his to get ready. I just have a curiosity. Can you compete this sort of place preference by putting something in the other compartment, like food or something, to fool the mouse?
Yeah, that's actually a really good experiment, and it's interesting because real-time place preference is like such an incredibly popular paradigm in the field, and nobody actually does that to look at like, does stimulation of that, like, does the primary reward induced by that stimulation, is it greater than the just a primary, like a natural primary reward and stuff like that. And so that would be a really nice experiment to do, and I may steal that from you.
Okay, great.
Yeah.
Some cereals. What is it? What kind of cereals they like that people always give?
Fruit Loops.
Cereals, Fruit Loops. All right. I guess Jean-Claude's picture is pretty much competing well against yours, Stephanie. So it's now Jean-Claude's turn. Fire up.
All right. Thank you, Paul. All right. So thank you also, Mario, for keeping the tradition alive, and thank you, Eve, for inviting all of us. So I guess, oops, I need to be able to find a way to get the slides rolling. Okay. So as means of introduction, I just want to talk a little bit about scales here there's a scales of connectivity, of essentially spatial scale, but these scales also reflect scales of investigation. So for instance, at a macro scale level, one can think of, for instance, at EEG levels or fMRI. And so these so these investigations are kind of somewhat constrained by their spatial scales of where essentially they're looking at. And then, you know, at the other extreme, you have the microscale levels, which really deals with individual synapse and so on. And we've clearly, you know, used optical approaches, and many others have used optical approaches to actually gain progress at this microscale level. And for instance, we've used two-photon encaging and calcium imaging to gain some progress on aspects of synaptic features or dendritic integration. But still, these investigations, these levels of these sort of experiments, they're still fundamentally constrained at the scales that they're operating at. So with this in mind, I think it's probably fair to say that the reason why optogenetic approaches have been so influential and have been used by so many of us has to do with their ability to scale, to bridge these levels of scales. For instance, you know, you can with optogenetic approaches, you can determine a number of synaptic features, get their influence at some of the mesoscale level, and actually bridge them all the way up to microscale levels. For instance, as Stephanie was talking about behavior, this is like quintessential microscale investigation. Now, historically, the other approaches that have been used to scale across these different, to bridge across these different scales are computational methods. They've been really good at being able to take parameters at one scale and make useful predictions at another scale. And this is one of the things that we've been trying to do over recent years, in part through collaborations. And today during the talk, I'm going to try to kind of convey to you on how we use these two methods in tandem to gain progress. So we've been interested in this over the years for a number of networks with different depth. So which essentially lies into the ability just to put in a channel rhodopsin in one region and actually look at synaptic features at a distant region. And I guess that the sort of the major question that animates us has to do with this is very simple cartoon. It's a very simplified way. You can imagine a neuron Z or a region Z that receives independent but parallel inputs from region X and Y. What is the nature of the computation that naturally arises from that particular network? We've used anatomy approaches, we've used uncaging approaches to get at that particular question and to understand what is the nature of the transform and what is the amount of information that is maintained and how is it transformed. Now, one way that we've done this too is as a conceptual testbed. We use the dorsal raphe nucleus, which receives inputs from a slew of regions in the brain. And we've chosen a bit randomly the medial prefrontal cortex and the lateral habenula. Both of them are the projections are glutamate, but they are so that you have an input that receives inputs from these two regions that are completely different. And so we thought it was a good test bed to actually compare what's the level of computation that could be occurring, what computation could be occurring in the raphe. So that's the sort of the overall sort of framework that we had. But perhaps in the spirit of this particular satellite that historically has at times been very given in terms of, or in sort of like technical workshops, the beginning of my talk, the first half of my talk is going to be really a generic description of classic optogenetic strategies. And it's going to be from our work, but it can be, it's very universal. And for these optogenetic strategies that allow us to infer some basic synaptic parameters and also connectivity motifs. And then in the second half of the talk, and then I'm going to jump a little bit, we're going to jump to serotonin synapse, where we've kind of applied and adapted these strategies to gain some insights into how we can study a monoaminergic synapse. And then in doing so, I'm going to try to give you a sense of how we developed over the years a computational formalism that leverages these experimentally derived parameters for network simulations to ultimately extract computations that are carried out by these networks. So this is one example. So one other thing that we do all the time is so you put in your channel rhodopsin in one region, you wait for a couple of weeks. This is an example of experiments have been in a neuron. You can get their spiking features with putting an electrode in there. And then by shining a light, you can actually test that your particular virus's batch works. We do have viral batches variability. Whenever we don't do this, somehow we pay the price later on. So it's good to test in a very tried and true system your particular viral batch that you have. And then so you can wait. And then the real thing is then we want to look at these inputs and how they influence their target region. So in this case, you make a slice of your target region, and in our hand, so you can see these are little dots. So these are axon terminals. They're axon fibers that are axes are orthogonal to your slice that you're making, and then this is just a little image showing these projection axons. And at least in our hands, you can make these slices, maintain it for about 4 or 5 hours. These isolated axons are perfectly healthy. And then essentially you just patch your neuron, and then you shine light, and then you get these responses. So far, so good. Now, so what can you do with this? In this particular example here, you increase, you shine light, and then you induce release. You get these synaptic currents. They can be blocked by AMPA receptors and NMDA receptor antagonists. So you can induce release of glutamate and activate these receptors. Now, this is mainly a diagnostic tool, but what can you do with this? Maybe you have a knockout, or maybe you have a particular behavior or some insult that you've given to the animal. So you want to be able to compare some features of your synapse between groups of animals. And then, so with these approach, with these little blue light represents here LED delivery, you can do the sort of classic synaptic parameters that most synaptic physiologists are really aware of. So you could look at AMPA to NMDA ratio to get a sense of the synaptic strength, or you could do rectification or decay kinetics of NMDA receptors to infer subunit composition. So as far as I know, these metrics have not been disputed in the literature, and you can certainly use them and to gain some insights into the parameters of these long-range connectivity. So one thing that you may want to be, that you may be interested in is, whoops, sorry about this. I'm going too fast here. Look at quantal size. So as you know, when you're classic electrophysiology, when you're stimulating electrically a bunch of axons, the amplitude of the current that you have is given by the number of synapses that you have times the probability of release times the quantal size. So you may want to be able to extract quantal size, but you don't know n, you don't know p. So how to do this? Then with optogenetic, you could use a trick that was developed in the mid-90s for just classic electrical stimulation by Chuck Stevens, where you replace your divalent calcium, you replace it with strontium, and essentially you're having a whole bunch of inputs of asynchronous events. These asynchronous events only come from optogenetically activated axons and not from the other ones. And then so you can have an idea of the quantal size specifically of the axon terminals that you are stimulating. Now, how to do that? So then so here this is example in normal calcium. You replace it with strontium. You have these asynchronous event. What we do is we sum everything in these raster plots, and above baseline, you're getting this population of events here. That these ones are only coming from those axons, and then you could do just do the basic stats and then that way you can infer synaptic strength. So we've done this with animals that had some treatments, and it's fairly straightforward and effective. So this is good. Moving on. one may ask, what about presynaptic release? So when you have these isolated axons, you're inducing channel rhodopsin-mediated currents in the isolated axon. Well, you do require action potential, even though that channel rhodopsin itself is calcium dependent, it's not enough. If you remove calcium, thankfully you get rid of the current, and then you also block it with a broad calcium channel blocker. So far so good. Now, can you modulate it by presynaptic classic presynaptic GPCRs? The answer is yes. The first thing that we've done here was with the canonical GABA B receptors that express just about everywhere. You activate GABA B receptors with baclofen, you shut down release. You could do this with other presynaptic GPCRs such as 5-HT1B receptors and cannabinoid receptors. This is cannabinoid receptors, you can activate them pharmacologically or with this very classic depolarization-induced suppression, in this case, excitation, and that works fairly well. So again, so far so good. It seems like for synaptic interrogation, optogenetic activation works as well as for electrical. But why is it going backward? Okay, let's go back to our favorite equation, A equals NPQ. What about probability of release? This is where it gets problematic. There's hints in the literature, but I think this is the best demonstration, or most explicit demonstration that's problems with release probability. This is an experiment that was done in Tom Ortner's lab a couple of years ago, where essentially they put channel rhodopsin in CA3 region and electrically evoked release or optogenetically evoked release. And then so here they've done this is quite heroic. They were doing paired recordings between individual CA3 and CA1 regions, which is the way to go, although the probability is very low. And so you're getting these pair pulse here, and here what they're seeing with electrically evoked, you're getting what canonically what yet you see at CA1 synapse, which is it's a facilitating synapse. When they were doing the exact same thing with light pulses, then they were getting a depressing synapse. And here this is really the kicker experiment. By looking at the coefficient of variation, presynaptically reducing release by light stimulation with adenosine, increased coefficient of variation. So this unambiguously shows that the reason why you had this is that the release probability was too high. So this is a real problem with optogenetically induced release, and that it probably has to do with the fact that the channel rhodopsin itself is calcium permeable and probably loads your presynaptic terminal with too much of calcium. Now, so if you would really want to know for a long-range connection, evaluate release probability, it is possible, but it's actually fairly challenging. So Carrie in the lab has worked quite extensively, at least in the hippocampus, with the glutamate sniffer, which is a circularly permeated GFP, which gets sensitive which increases fluorescence when it sees glutamate. And then this is an example. This is a spine here with a piece of dendrite, and then you can electrically evoke release and monitor the glutamate sensor signal at single spines, and then you can partition the glutamate the sensor signal with successes and failures and extract a number of features. We've done this in the past, where here in this paper, Carrie looked at synaptic release dynamics. And with Richard No, we published what I really like, a Frontiers paper, where we used that approach to extract a number of release features, including the number of individual vesicles released at these synapses. So in theory, you could use this with optogenetic. And so the way to do this is, say you have a whole bunch of axons. Some of them are labeled with channel rhodopsin. Here, this is a mistake. Channel rhodopsin will not work. You have to use a red opsin for this. So you're inducing release, you're identifying by the glue sniffer signal what are the axons that are activated, and then you simply electrically activate them. You can activate a whole bunch of axons, you really don't care, but then you only monitor the glutamate signal from those ones that were lighting up by optogenetic. This is an example. This is a serotonin. These are serotonin neurons. and then the green dots reflect glutamate sniffer that were activated when the crimson was activated optogenetically. So you can identify of all the synapses, you can identify the ones that are optogenetically released, and then you just put in a stimulating electrode and monitor that particular one. Now, I need to be fully candid here. That approach is feasible, but we certainly have not been able to get it to work at a high throughput level. These are very challenging, but at least if for whatever reason you really want to know the release probability of identified long-range circuits, this is at least one possibility. Now, you may want to, you may be interested in small-scale connectivity. This is a classic approach. If you have a synaptic current, when you shine light, and then you want to make sure that that current comes from the activated, only from the infected axon that comes from, a mile away. The trick is what you do is that you do what we call the TTX4AP trick, which is kind of classic in the field. And then so you can monitor here, like the hypothetical axonal response is that when you have channel rhodopsin activation, you get a calcium threshold, you get sodium, spike. When you put in TTX, obviously you remove action potential. So your depolarization induced by channel rhodopsin does not work. When you block the delayed rectifier, essentially you let the depolarization go unabated, and essentially monitor or trigger a sort of nasty looking calcium channel opening. And then so you're getting a monosynaptic current back. So this is one example. You're getting EPSCs, you block it down with TTX, and then you rescue it for 4AP. Now, because of this calcium channel spike is not as temporally precise as the regular spike, you will expect some temporal deformation of your inputs, and this is exactly what you get, and this is what you expect. Now, a word of caution with this approach. It's very sensitive to false negative. If you do not get your rescue with 4AP, you can still have your a direct monosynaptic activation. But however, I don't think it's prone to false positive. If you did get your rescue, even though partial, I think you can take that result to the bank. Other connectivity feature that you may want to know is so your long-range connectivity or your long-range pathway, what type of motif does it trigger? So here there's an example of recordings from the Raphe nucleus where the channel rhodopsin was done in the PFC. You depolarize your cell to about -30 millivolt, and then you can get this mixture of EPSCs and IPSCs. If you do the exact same thing from at this synapse right here and topinuncular to the habenula, you get essentially the same thing. So how do you discriminate the two? Well, this is the answer. This one is a feed-forward inhibition. Whoops. This one is a feed-forward inhibition, and this one reflects co-release of GABA and glutamate that was shown this is some of our recordings, but that was previously shown by Raul Malino's lab. So how do you discriminate the two? Well, I told you that the release probability of channel rhodopsin mediated axons is too high, and then when you have an interneuron in the middle, so you're going to get you can make a whole bunch of predictions on the variability of that current versus that one, and also the temporal jitter that you get in time. So for instance, this is one response that you may get with your channel rhodopsin in these inputs. You depolarize to -25 millivolts and you get the two. You can do a very simple analysis of your jitter in time and in amplitude. So for instance, the feed forward is going to be far greater jitter than your direct monosynaptic current. And then, so this is exactly the type of temporal behavior that you would expect with a feed forward motif, which you'll not get with GABA or glutamate co-release. And then you could also do the final nail in the coffin, which is by doing just simple pharmacology. If you block all of glutamate drive in both of these ones, you can essentially shut down everything, and this is what you have. And if you follow the TTX4AP strategy, this strategy says that you will rescue this one and you'll never rescue this one. You will rescue the EPSCs and you'll never rescue the IPSCs. And this is exactly what you have. So these approaches, like this, provides a pretty strong formalism to look at and determine these connectivity features. And we've done this in the RAFE, and including with some from neuromodulation effects. So essentially what we showed is that you have the axons from medial prefrontal cortex activate directly monosynaptically serotonin neurons and a feed-forward activation. Now, there was cannabinoid receptors in there that showed target specificity. In other words, you have a common axon from a common region, common population of axon. These guys had a lot of cannabinoid receptors, these ones didn't. So when you're in a condition of elevated endocannabinoid, essentially you shut down the feed-forward, inhibition, and you favor the monosynaptic activation. So Sean Geddes published this and a couple of years ago, and Emerson Harkin just published a bioRxiv paper. If you're interested in what is the computational meaning of shutting down a feed-forward motif in this particular region. Okay. So I've talked about mainly glutamate synapse. In the RAFE, we're going to stay in the RAFE, but I'm going to switch gear a little bit and talk about serotonin itself. So the RAFE here is a relatively small number of neurons that projects to the entire brain. And then you can see here, you can get a visual representation of the extent of the arborization. This morning during the talks, there's a lot of interesting ideas about neuromodulation and how it modulates brain states, and serotonin is certainly one of them. If you look at textbooks, the textbook knowledge on serotonin in the RAFE has to do with this particular business of autocrine somatodendritic serotonin release model, which has to do with when serotonin neurons spike, they release so-called somatodendritically serotonin, and that activates these inhibitory receptors. And this is very dominant model in the field. Long story short, we've put in electrodes in the RAFE now for, I don't know, 7 or 8 years, and we've looked at predictions of this model, and we were never able to find any support of this. So with this in mind, so then we set out to use the optogenetic strategies and that I described before, and to look at connectivity features in the RAFE. So real quick, so we use a serotonin transporter Cree line, and then you use a Cree-dependent channel rhodopsin or variant. In this case, it's cheetah. You can validate it well. So you put an electrode, you spike, you shine light, and then the neurons spike. Now, what we did after this was we found a way to, through kind of a hard work actually, find a way to get a sparse transduction using the viruses in the RAFE, where essentially you would have a whole bunch of serotonin neurons, some that were expressing the opsin, some that weren't. And then essentially you could just shine light while patching either red cell or either green cell. Now, if you're patching the red cell, which does not have the channel rhodopsin, and then you shine light, essentially you're implementing an optical analogy to just simple paired recordings, which was used in the cortex forever. And actually this is the gold standard to show that cortex is a recurrent network. And when you do this approach, essentially what we have, we shine light, and then we get these hyperpolarization in current clamp, and then these outward current and voltage clamp. And that current rectifies, so it's mediated by a GERT channel, and is blocked by 5-HT1A receptors. So essentially what this experiment shows, and we have other support for this as well, is that there is serotonin to serotonin synapse in the RAFE, and the RAFE is organized in a recurrent network, which is completely different than at least model or at least the textbook knowledge of the RAFE. So with this in mind, then maybe we have a way to study the serotonin synapse. So then you can do a whole bunch of parameterization of this, and we were really interested in very basic mechanisms. So is this synapse a deterministic synapse, or is it a probabilistic synapse? And one way, it's a little bit complicated because this is a very long, protracted current, and you can't really do the sort of classic experiments that one would do, say, for a glutamate and GABA synapse. But still, you could take a basic binomial formalism and just say, well, if it's a deterministic synapse and the variability and the probability is really high, then the only variability that you can have of your current is going to be given by the variance of your measurement. If it is a probabilistic synapse, then there's going to be some extra level of probability of variance, and this you can show this by this very, very simple numerical simulation, and then you get with the probability of release the variance. The relationship between probability of release and variance shows a nice hyperbolic relationship. So if any movement from along this axis of release probability of variance shows that you have a release probability of less than 1. So we've done this with this particular synapse, and essentially what we found was that the variance did not change at all by increasing pulse number. And that, with a number of other evidence, shows that the serotonin synapse is deterministic and quasi-linearly encodes firing rate. And this is kind of the big deal because synapses don't do this. They don't encode firing rate, at least not linearly. But then what Michael in the lab was, well, is this real? And so then essentially he went back to this very classic method of just tried and true electrical stimulation. And through some work, he was able to convince himself that he could actually recapitulate this and do this. But then by looking really carefully, he said, you know, wait a second, there's successes and failures. So I don't think this is a deterministic synapse. And then he started to dabble with this and do single pulses and paired pulse and so on. Which really kind of says, well, you know what, this is not a deterministic synapse, it's a probabilistic synapse. So then what he set out to do is, okay, let's do an experimental parameterization of release dynamics at this synapse. So he started to do single pulse and pair pulse at different stimulus interval, and did a whole bunch of a bit weird experiments. We could go through this, but this is only going to be numbers. So then the trick would be to take all of that and feed it into a model and try to capture that release dynamics. And right, our next door neighbor, Richard No and Catlin Toth, they were collaborating in a linear nonlinear model to capture a synaptic dynamics in this particular case at mossy fibers in CA3. So essentially what Michael did is that he adapted that model, but to a GERD kinetic. And lo and behold, this worked extremely well, and he was able to have a perfect match between experiment and model, and including the model was also able to capture stuff that he's never seen before. So now we have a nice model to capture that weird monoaminergic synapse. So the DRN is organized in a recurrent inhibitory network. The synapse we have, they activate over a protracted time scale, and they have complex dynamics. So what? So what do we do with this? So then we went to further modeling. So Michael's idea was, okay, we have this recurrent network. What happens when this recurrent network is perturbed by some inputs coming, for instance, from the habenula? And then here, the basic here is just to focus here. So you get this GERD conductance, and as you perturb, you increase the perturbation rate, then you're increasing your GERD conductance. So then we validated this experimentally. So we put the channel rhodopsin in the habenula, and then look at neurons in the DRN when you're giving habenula input at different rates. So 1 hertz, you get these nice synaptic current, and then when you push a little bit, then you get these synaptic currents, which is followed by this outward current hypopolarization, which is mediated by this 1A current. So then he did even more modeling, and then this was smelling a little bit of input hierarchy. So essentially what he did is that, okay, let's model this recurrent network. Let's have habenula inputs, and let's have some generic other input. In this case, area A. The truth of the matter is this was modeled against the PFC inputs. And then so you can have the PFC inputs that come in, and then what happens with all of the features that we've experimentally derived, what happens when you challenge the system with an input at 5 hertz, what happens when you do it with an input at 20 hertz. And essentially what you end up doing is the 5 hertz doesn't do much. These are the individual raster plots of the population. And then once you start to push a little bit the frequency, then you can shut down completely serotonin response. So this had a prediction in that, so we know that the habenula signals threat. So the idea is if you have serotonin output with increasing threat, so the small threat will increase the output, whereas you get to a point where you recruit this 5-HT1A recurrent inhibitory network and you shut it down. Real quick, so we wanted to, only 3 slides left. Am I good in time, Paul?
30 seconds.
All right, so we're good. So what Michael did is the idea is that serotonin probably mediates, there's this nice theory where serotonin mediates persistence in a given behavior. So what Michael did to play with this idea was he developed a head-fix classical conditioning task where the animals learn to associate different cues with a reward. Now the reward is given with some jitter, so that the animal knows when he's going to get a reward with some frequency with tone presentation, but he doesn't know exactly when. So this generates anticipatory licking. So the animal gets the tone and then will start to lick in anticipation. And this is one example here. So you can monitor the jaw here a little bit, and then he gets the tone that he knows that it's going to get some reward, and then at the reward he kind of pushes forward to try to get it. Now we do the exact same thing, but during the trace period, right during when the animal is expecting to get the reward, we activate the habenula input, and in the raphe, and then you see the animal completely stops licking. And so long story short, this is example. So you get a lick rate, you get the time from the tone. So the tone is given here, the reward is given there. This is the trace period. When the animal gets the tone, licks in anticipation. When you activate at 5 hertz, nothing happens. When you activate that at 20 hertz, which is completely in line with the model and the experiments, the slice physiology experiments, completely shuts down the licking behavior. So right now, Seb has developed a way to image in the lab. So Sebastian, what he did is that he implants Grinlands in the medial prefrontal cortex, and then he generated this nice stereotaxic apparatus where he can adjust the pitch and the roll to align the Grinlands with the objective, and then we can get nice Castle imaging. And we're dabbling now with a novel serotonin sensor to be able to monitor serotonin dynamics during these tasks and other sort of foraging dynamical tasks. So in conclusion, so I think, so what I was trying to do is to describe a formalism where we generate a dialogue between computational approaches, electrophysiology, and optogenetic approach. Now, real quick, we show that the raphe is organized in an unsuspected recurrent inhibitory network that acts on protracted timescales. And we've been working to try to identify the neural computation that such a recurrent inhibitory network may support. And if you want to speculate a little bit, it makes perfect sense that when you have an intent to do something, for instance, if you're a mouse and when you want to eat berries in the open field, you constantly need to contrast that internally generated intent with some features of your environment, and if you have a little bit of a threat, maybe you want to do that intent faster. But when your threat gets to a certain level, then you may want to just forfeit that reward essentially in a go no go binary outcome. So either you go or you don't go, but you need to parameterize some linearly represented threat signal. And I think we found at least one neural mechanism that could support this binarization. So that's my last slide. So I need to thank Sean, Michael, Emerson, and Sebastian, and Celia, who've been involved in this work. And all of this has been done in collaboration with our computationalist extraordinaire, Richard. No.
Great, thank you, Jean-Claude. That was awesome, terrific stuff. Maybe you should pair with Stephanie to combine threats with rewards and see which side does the mouse go. We have the primates coming, so that's one concern, and they're coming in 7 minutes. And so if there are questions, we can take some questions. If some of you need a coffee on the West Coast, Mario said short break, doesn't say coffee, but I guess on the West Coast, you're still allowed. On the East Coast, you gotta weigh in the sleep problems tonight. But we can chat until then. And those who want to go, just go and come back at 4. Is that fair, Mario?
It is, absolutely.
So I just have one technical question, Jean-Claude. I'm sort of interested in this glue sniffer stuff. But I was a bit worried that for measuring something as sensitive as P, probability of release, are there any technical concerns about the dynamics of the transmembrane protein on the surface of the postsynaptic cell? Like, does it diffuses in and out of the area where you're measuring? Is there some bleaching concerns?
Yeah, that's a good point. So actually, that's exactly we've written. It's funny. It's one of my favorite paper that we've ever published. Is a Frontiers paper where we cover all of that. There's a lot of signal processing in there. There's calibration of the glutamate sniffer with uncaging. So. And essentially, we compare the sniffer with glutamate, with calcium imaging through NMDA receptors, and we pay close attention to the temporal dynamics of the actual signal. And it turns out that, so say when you're uncaging and you're monitoring your AMPA-mediated current, say, of course it's a mile away at the cell body, but still you can get a pretty good measure. Turns out that the sniffer is really darn good. The dynamic range of detection is pretty close to that of an AMPA receptor. The affinity is a little bit higher, so the affinity is actually, if anything, is closer to an MDA receptor, but have the kinetics that are close to an AMPA. It's a little bit sluggier than AMPA, so when you push when you push the frequency, you do get in trouble. You may want to deconvolve a little bit your signal, but over a pretty broad sort of frequency range, it's pretty good. Now, the real issue is, which is for everything, is signal to noise ratio. And it actually comes, so long story short, we're actually not even sure that we can discriminate a signal to noise ratio of failures events for an AMPA mediated current. In that paper, we kind of show quite well that we can get sniffer events at single synapse that are not accompanied by AMPA receptor mediated currents. So if anything, the sniffer signal is a little bit more sensitive than AMPA receptors to detect them. So there's glutamate being. I'm not talking here about a glutamate silent synapse. This is a fully functioning synapse, but the variability of glutamate in the vesicles that are given to the synapse. Some vesicles have very little glutamate, and then when they're released, they don't trigger any AMPA receptor mediated currents, but yet we can see them with the sniffer. Oh. And it's part of that variability that we've used with maximum likelihood detection methods to be able to extract. One of the things that we try to extract is the number of vesicles being released. And that number of vesicles is dynamically regulated. When you push the frequency, you start to get a little bit more vesicles. So I don't think that anybody now believes that Korn and Faber were still right. I think at most synapse we are getting more than one vesicle being released, but we've certainly used that to provide a bit more weight to that idea. And our numbers are about 2 to 3 vesicles when you have a full-blown release.
2 to 3. All right. anyone has a question? There's 2 minutes left before.
Oh, I see Ed had a question.
I don't see.
Okay, the false appearance of, yeah. So Ed was asking when we were optogenetically inducing release of serotonin, it seemed to be like it was a deterministic synapse. And yes, you got that right. And the reason is, we think it's the same reason why release probability by optogenetics is exaggerated. Essentially, because it was a GERT mediated current, we had to go, to jump through hoops to look at that. But we reached the same conclusion. When you study glutamate release or serotonin release by optogenetic, release probability is too high. And it is a major, depending on what you want to do, for I don't know, 80% or 90% of what people use is fine. But if you are interested in release probability, be it serotonin, be it glutamate, probably be it GABA, it's a problem.
So I see that Keith is here and he raised his hand. Is that to say, hey, it's my turn to talk, or is it because he has a
No, I just had a quick question for JC. You mentioned at the end there working with a new serotonin sensor.
Yeah.
Is this something that's genetically encoded, or is it a chemical? Or
yeah, it's a genetically encoded. Yeah, we have two in the labs. We've just got them recently, so I refuse to answer, to commit to anything without my lawyer at this point. No, we just started.
Stay tuned for next time, huh?
Yeah, something like that.
Okay.
All right. Question and answer are over. We have to move on to the next session. Thank you, Stephanie. And thank you, Jean-Claude. That was really great. And I'm going to pass on the microphone to Martin Parent.
Oui, bonjour. Merci, Paul. Just for those who don't know me, my name is Martin Parent from Laval University. I work at the Cerebral Brain Research Center in Quebec City, mainly on Parkinson's disease, basal ganglia, looking at how microcircuits reorganize in this pathological condition using non-human primates and also mouse model of the disease, as well as post-mortem human brains. So it's my great pleasure to chair this fourth session on non-human primates and human disease model. Okay, so, well, you all know about all these fancy and powerful technical approaches, such as optogenetics, CRISPR-Cas9, calcium imaging, and so on, that are commonly used in smaller animal models. However, applying these same approaches in larger animals, such as the non-human primates, has been a real challenge. So, of course, this is deeply needed if we want to better understand the primate brain. That, I think, shows a higher level of complexity, and maybe eventually transfer some of these approaches towards potential clinical use to treat human diseases. So I'm a little bit biased, but I'm sure that this session will be the most exciting one. Not because of me, of course, but because we have two great talks scheduled. The first talk is from Keith Murray's lab, located at McGill. And the first part of this talk will be given by Jean-Bastien Bott. He is a research associate in the lab. And Jean-Bastien will talk about calcium imaging in mice and non-human primates. And then we will hear from Keith Murray himself on astrocyte imaging in marmoset. And the second talk will be given by Maria Sapers and Ellen Koch, both from Lynn Raymond's lab at UBC. And the title of the talk is Using in vivo optogenetic sensors to elucidate corticostriatal dysfunction in mouse models of Huntington's disease. So I would like, again, remind everyone to keep your microphone muted, and if possible, maybe your camera on, so it's more enjoyable for everyone. And I will take questions in the chat and ask them after the talk, or you can ask them yourself by raising your hand after the presentation. Please, speakers, stay on time. You should aim for 25 minutes presentation so we can save time for questions. So let's begin with Jean-Bastien. If you can share your screen, Jean-Bastien. Yeah, and maybe go full screen.
Can you see my screen?
Yeah, the floor is yours. Everything is fine. Thanks.
Okay, great. So thanks very much for this introduction and for the invitation to speak today. So today I'm gonna illustrate how 3D behaving calcium imaging is a very good tool to better understand the relationship between network activity and memory processes, first in mice, and then why it should be interesting to investigate this in non-human primates, and especially in marmosets. So in the lab, we are interested on investigating what is the relationship between the cognitive map and memory. So basically, the hippocampus is well known to contain play cells. So those are cells that are active in a specific portion of the environment. And together, those cells are understood as a system that might be the substrate of Tolman's cognitive map. So basically, if you take the activity of all the cells from the hippocampus, you're supposed to have a nice representation of the whole environment. So to investigate if there is a relationship between this cognitive map and the memory, we decided to use GCAM6 fast calcium imaging using miniscopes, and especially the open source miniscopes from UCLA, because that allowed to track the activity of hundreds of neurons across days. So basically, this procedure requires a three-step surgery. So first, we inject the GCAM6 in the area of interest. Here it's dorsal CA1. And as we would like to target only principal cells, we use the virus under the control of KMK2 promoter, which allowed to target specifically principal cells. So 2 weeks after, we implanted a 1.8 millimeter diameter green lens over the C1 region. And 3 weeks after, we do a base plate surgery, which consists to implant a base plate where we will plug the miniscope over the head of the animal. And then we go for behavioral testing. So first, we started with a pretty simple task, basically a curved linear track that have two different reward regions. And water restrict mice will just have to run from one end to the other end of that linear task. So it's a kind of simple task. There is not a lot of cognitive load in that task. It's simple. The mice just run forward to find the reward. And here is a maximal projection of the neuron footprints that we can get from one example mouse. And the footprints from day 1 are shown in red, from day 2 in green, and from day 3 in blue. So you can see that there is some cells, especially the ones that appear in white, are cells that are active for all the 3 days of testing. So of course, we are recording CA1 dorsal hippocampus in rodents. So we get a number of very nice looking play cells. So here's an example of a play cell that is active in both directions, in both running direction, and that show a stable place field across the training during that day 1. So given given the use of the calcium imaging, we can track those neurons across days. And that neuron, for example, do show a shift of its field of activity, meaning that these neurons have a very low intersection correlation. There is a remapping, the field of activity of that neuron is shifted to a new place. On that other example, we have a neuron that is much more stable, keeping its field of activity specific to one direction of running, which is currently seen in a linear environment. And that field of activity remained pretty stable across days. So then the question was how to measure the cognitive map stability across days. So of course we can have a look on all the neurons independently. We can classify the neuron using arbitrarily determined threshold as place cells, non-place cells, and look on how they are stable across time. But we did prefer to have a more inclusive strategy, just taking the activity of all the cells that were detected to compute a population vector, and especially population vector correlation between days. So basically for each spatial bin of the environment, we will compute for all neurons the average frequency of activity during day 1. And for the same spatial bin on day 2, we'll do the same, and we will cross-correlate those two population vector. And if we get a high correlation value, it means that the representation of the environment stays stable. And if the correlation value is low, it means that there was a remapping. So here's an example from the same mouse between day 1 and 2. So as you can see, there is one big red zone here with high correlation value, meaning that here the population vector, the cognitive map was stable between those 2 days. But on other region of the maze, there are very low correlation value, suggesting that there was a remapping processes for those portion of the maze. So we did those calculations for 5 mice for a total of 5,288 cells across the 6 days of training. And here is the population vector correlation that we got. So basically, on the first day, the population vector are not very strongly correlated with subsequent day, suggesting that there is a remapping processes. On day 2, those processes seems to be even more drastic because the correlation value are lower on subsequent days. But then from day 3, it appeared that there is some stabilization of the environment representation, and especially for the reward region. So it appears that in those simple tasks, there is some cognitive map stabilization across days, but the stabilization seems to be restricted to some portion of the maze, and especially to the region where the mouse got a reward delivery. So this task is really simple. So then we wanted to investigate what occurs in a learning task, because we know, of course, that okay, there are place cells in the hippocampus, but the hippocampus is also known to play a major role in memory, and especially in spatial memory. So for that, we use a dry star maze task. So basically, the mouse is put in one of 4 pseudo-randomly selected start arm at the beginning of each trial. It stays for 30 seconds in that start arm, and then the door opens. The mouse freely explores the maze, and using allocentric cues, the mouse progressively learns to locate which arm contains the water reward across days. So we do 16 trials of training per day during 3 days. And as you can see here with the % of direct trajectory to the target, across the 3 days of learning, mice reach better performances. And on the 4th days, we conduce a probe trial with no water delivery, just looking on how the mouse investigate the maze. And as you can see, there is a strong preference for mice to visit the previously rewarded target arm, suggesting that indeed those mice have formed a strong spatial memory of that context. So going back to the cognitive map stabilization, we found that compared to the linear track, the cognitive map is much strongly stabilized in this learning task, and especially for the start arm region and for the reward region. Also, the stabilization of the cognitive map is not homogeneous, so it's restricted to the start arm and to the reward arm, but you can see that there is some progression, and with more and more stabilization in the choice region right before the entry to the reward arm. So of course, as we also found a stabilization in the reward region in the linear task, we were particularly interested by understanding what occurred in the start arm waiting period, because those regions of the maze are really, really stable since the first day of training. So here is for each individual trial sorted by the start arm location. So -1, -2, +2, and +1 in relationship to the target location. You can see that the population vectors show a strong correlation when for the same start arm region, but low correlation for the other low start arm location. And that seems to be more and more clear across the training. And using support vector machine decoding, we show that this activity is sufficient to accurately decode the start arm location. So we found in conclusion that the cognitive map stabilization is indeed required for learning and memory processes, but the stabilization is selective to silent region of the maze. So the reward region in both tasks, but also the start arm waiting period and the start arm, sorry, and the choice point in the maze. So now the question is, okay, what would we want to test that in primates, and especially in marmosets? So marmosets, as you probably know, are small New World monkey that are decently small, and but that are much closer to humans than rodents, and meaning that as a model they are much better treated because they have a more similar immune system, pharmacokinetics, sleep patterns, etc. Also, they have a complex behavior that are really, really close to humans, especially for visual and language processing, and also due to their prosocial behavior. They form the family units with cooperative raising of young and parental cooperation for the development and vocalization, for example. And as a model, the good thing with marmoset is that they have a short reproductive cycle that give you twins or triplets, but the triplets are generally, one of the pups is generally non-viable. But every 6 months you can get new animals. So the main advantage of marmosets is that they have a primate brain. So here in comparison to a mouse section, you can see that the brain is much larger first, and also it's an advantage, but it can also come with some complication. Is that as a primate, the hippocampus shifts ventrally compared to rodents, meaning that the distance to travel to reach the hippocampus is much larger than in mouse. So also in terms of model, the good thing is that they have some human-like pathologies that occur naturally, like amyloid beta accumulation with aging, which is not found in rodents. So potentially that marmoset can be a very good models for sporadic Alzheimer's disease, for example, because in mice for the moment, we basically have to put 5 familial dominant mutations to get something that resembles Alzheimer's disease, which is not a good model because, of course, for 99% of the patients, we just have a few genetic links like APOE4, for example, that doesn't really induce a pathology, but maybe in marmoset that would be more close to what we can find in humans. So also the thing with marmoset is that they have a very important, as primates, they have a very important visual system, meaning that the activity in the hippocampus might be different from what we found in rodents. For example, rodents are characterized in the hippocampus by place cells. Place cells, it's cells that are active in one portion of the environment that is occupied by the animal. it has been found from Julio Martinez's lab recently that the marmoset, here it was not marmoset, it was a macaque, but it's probably the same in marmoset. The place cells are not really coding for place, but more for gazing. So for when the animal look in a particular direction, there will be a place-like activity instead of coding for the exact place where the animal is in the environment. So the sensory dominancy of the information coming to the hippocampus may actually change the activity of the principal neuron from the hippocampus. So it's important to investigate that in non-human primates because to better understand what can occur in human hippocampus.
Jean-Bastien, I'm sorry to interrupt, but do you leave time to your boss? What's the plan?
I have
everything is fine on your side.
So yeah, just that's just to say that we are currently starting. So we did inject the first animal in Julio Martinez's lab in London, and those animals will be implanted in the following months. So we are really looking forward to see what occurs for the cognitive maps in primates. So before giving the floor to Keith, I will to thanks Sylvain Williams because all the mice work has been done there. Of course, Keith Murray and Julio Martinez, which is our collaborator for the marmoset testing part of this project. Thanks for your attention.
Thank you very much. Keith, the floor is yours. I won't take too much time to introduce you because the time is running.
Okay, great. Yeah. Let's see here. Can everyone see my slide there?
Yep.
It's okay. Great. Well, thank you very much, Martin, for the nice introduction, and thank you to Mario and Yves for the invitation to come and present here today. You know, JB gave a very great presentation about how we can learn a lot in rodents in terms of understanding behavior, but also the physiology and the neural circuits that underlie the behavior. But there are some questions that, you know, we need to ask in higher order species to maybe understand whether or not there are differences between species and whether or not the mechanisms and pathways are the same or different. So I just have a few minutes here, so I'm not going to spend too much time. I'm going to go pretty fast. But in addition to our interest in neurons and their physiology and their activity patterns, we also have an interest in understanding glial cells in the brain, you know, as other important cells in the brain that help contribute to the entire function of the brain. And over the course of today, we've really got to see, you know, how neurophotonics and optogenetic tools can be used to get at, you know, the properties of brains at different levels, at different scales, as JC mentioned as well. And how we can use different model organisms from invertebrates all the way to, say, non-human primates and human tissues to understand how the brain is organized and how it functions. So today's goal, I'm gonna be really quick because I don't have much time. And so I thought I'd just pick something very easy to talk about for the remaining minutes of the session here, and tell you something about our interest in astrocytes in both the rodent brain, but now the non-human primate brain, as well as the human brain. And I'm going to just briefly tell you why we're interested in astrocytes and discuss why we're studying astrocytes in the marmoset model, and highlight some recent work related to understanding the properties of marmosets in the cortex, hippocampus, and cerebellum. In particular, kind of taking a step back, prior to doing optogenetic work and calcium imaging, is just to understand the basic features of astrocytes in the marmoset brain. So this is looking actually in the rodent brain. And again, what you see is just how complex an astrocyte can be. And this is just looking at a piece of an astrocyte, you know, where you have the intricate anatomy of the cell. And this is where we've reconstructed about 638 serial sections. There's about 500 PSDs in this particular area. And you can see the astrocyte in yellow nicely interacts with synapses and the vasculature. So astrocytes really are probably master regulators of a lot of information. So they're integrators of information. However, we're just getting to know what these cells actually do in the brain. So why do we need to know more about astrocytes? Well, they're structurally very complex. They interact with a lot of different cell types, neurons, oligos, microglia, and they have a lot of interesting molecular properties that control the physiology of neurons and the circuits. They're strategically positioned really things, including ion homeostasis, neurotransmitter homeostasis, neurovascular coupling. And on top of that, they have some really interesting calcium properties, calcium elevations, something called calcium microdomains in the periphery of the cell, but also large types of calcium events occurring, for example, near vasculature. And as you can see here, this is probably related to neural activity, but also metabolism going on in the cell, and the ability to respond to say, synaptic activity and maybe redistribute energy substrates throughout the parenchyma. Astrocytes now are known to function a lot of different roles in the brain, for example, in breathing, appetite control, vision, olfactory memory, and sleep. But another interesting property about astrocytes is that they have this kind of dual side where when they get upset, they take on some really strange properties, and that can be either productive for brain repair or detrimental for brain repair. So really understanding the properties of both the healthy and the diseased astrocytes is going to be really important in understanding brain function, but also recovery, for example, in disease states. So as JB went through the strengths of the marmoset model, and I'm not going to go through those again because they're pretty clear. There's some really interesting strengths of the marmoset brain and behavior of the animal and cognition and how its experiences and things like that when bred in captivity. But another thing that needs to be highlighted is that there's some new resources that are becoming available to understand marmosets and the marmoset brain, but also the ability to do genetic engineering in marmosets. And so that's for a separate talk, but the interest is in the future for us at McGill to establish transgenic models and genetic models that we can use to mimic human disease. And so that's potentially maybe for hopefully next year I can give an update on that. There's a lot of investments by a lot of different universities, agencies, institutions towards marmosets these days in terms of using it in various ways, especially for modeling neurodevelopmental disorders as well as neurodegenerative diseases. So as JB mentioned, there are differences between rodents and humans at multiple levels in terms of the brain, in terms of the individual neurons, the molecular patterns of the neurons, and the physiology of the neurons. The same thing also applies to astrocytes, but we know much less about what's going on with astrocytes in the primate brain. So I'm just going to spend the last couple minutes just to tell you about some of the work we're doing, just to take a step back and to understand the properties of astrocytes in the marmoset brain prior to doing any form of, say, calcium imaging or optogenetic manipulations. And so this is a study that was recently completed. It's resubmitted again from my postdoc, Yorka Munoz, and where she did a nice kind of systematic evaluation of marmoset astrocytes. And just to understand what kind of markers do they express, what do they look like, and how are they organized? And so she did a really complete study. I'm not going to go through all the details, but one of the interesting things that we found is that many of the markers that work well in rodents work quite well in marmosets. In fact, some of them may even work even better in marmosets than in rodents. And as you can see on the lower left there, there's really strong conservation of antigens that for human, rodent, and marmoset astrocyte proteins. And because of that, many of the antibodies that we use in rodents can nicely be used in the marmoset brain. So in this study here, we used 3 different marmosets, 2 males and 1 female. Just note that these marmosets were were perfused or euthanized usually for other health-related reasons, but so that allowed us to do a full descriptive analysis of astrocytes. But just suffice it to say, there's some really interesting patterns of markers in astrocytes. However, markers like glutamine synthetase generally do well at marking all astrocytes in the marmoset brain. And we use this as a way, for example, to tag all astrocytes and then do some form of hierarchical clustering analysis where we can basically look for different patterns of gene expression of various markers. An example of this is the gene KIR4.1, which encodes the inward rectifying potassium channel, which is known to show variation in expression in the mouse brain. And what we found is that in the marmoset brain too, this is also the case. However, other markers, for example, against the glutamate transporter EAT2, there's less variation in expression and less heterogeneity in expression. There's some interesting features of marmoset astrocytes that I don't have time to get into, but marmosets also have this mysterious interlaminar astrocyte that is found in layer 1 of the cortex and extends long processes into the deeper layers. And no one knows what the function of these cells are. These cells don't occur in rodent species. So this is, one really noticeable difference between the marmoset and the mouse brain. There's also some really recognizable differences between the rodent and the marmoset with respect to polarization of astrocytes in the hippocampus. And I don't have time to go through it, but there's some really interesting features of astrocytes, for example, that sit at the interface of the stratum radiatum and the pyramidal cell layer, and with a really strong polarization pattern. I'm just going to skip to the very end and just say, hopefully, this talk was just a primer about what we can do maybe with understanding astrocytes in the brain and using the marmoset model. And new tools are coming available almost every week or every month that could be applied to marmosets, including new adeno-associated viruses that can be used to specifically target certain cell types in the marmoset brain. And with that, I will just thank Yorka Munoz, the postdoc who did the immunolabeling work, and work with Francisco Cuevas Pacheco, who helped with the cluster analysis of the astrocytic markers, as well as Sylvain Williams and Julio Martinez Trujillo for collaborations. Thanks.
Thank you very much, Keith. A wonderful talk. We have time maybe for one or two quick questions. I have one from Ed. They truly are the stars of the brain. Is there any evidence for these interlaminar astrocytes in other primate brains?
Absolutely. So human brain, humans have these amazing interlaminar astrocytes. Other non-human primate species as well. Apes as well as humans have even more elaborate interlaminar astrocytes that are but have unknown function at this point. So it would be quite interesting to be able to track say, for example, the calcium activity in these cells, for example, in motor or sensory cortices, during activities, during certain types of behaviors, to see how they might be integrating information and why they need to project from the superficial layer all the way to the deeper layers. Thanks, Ed.
Another question from Arthur Kanya. Wow, Keith, I didn't really catch how you are doing the astrocyte classification. Is it by co-expression of other markers or laminar location?
Yeah, thanks, Arthur. We're doing both. So we're using glutamine synthetase as a general marker for astrocytes. GS and SOX9 generally label every astrocyte that we've seen. And so we can use it as a way to create ROIs around astrocytes and then go in and measure the signal intensity of the various other protein, for example, KIR4.1 or glutamate transporters, and then look at the signal variation using agglomerative cluster analysis. And we're able to do this in different regions of, say, cortex. And so we can do the cluster analysis, identify how these astrocytes kind of sort out into groups, and then remap these astrocytes back onto the cortex to figure out where they're positioned. And so we're starting to learn how they're grouped together and how they might be organized in a lamina specific manner.
All right, thank you very much. Nice talk, guys. We will move on with the second talk now. So if you want to start sharing your screen.
Sure.
So this talk is from two lab members from Lynn Raymond's lab at UBC. We will begin, I think, with Maria Sapers. So the floor is yours.
Thank you. So I'm just a research associate in Lynn's lab, and I'm going to be talking about a lot of people's work that I absolutely cannot take credit for, and also about our collaborations with Tim Murphy here at UBC, who does the coolest things, and we've really benefited from having him around. So Lynn is a neurologist, and she gives us the clinical perspective on Huntington's disease, and it really helps to focus our research and think about the impact in the clinic. So okay, Huntington disease is caused by a mutation in a single gene, which is the Huntington protein, and it has this repeat region, which is a CAG repeat, and in mutant Huntington protein, it's expanded. And the length of the expansion influences how severe the disease is in people. So it not only presents with motor symptoms, but patients also have psychiatric symptoms, and they also show cognitive dysfunction. And we've known the origin of the disease for a long time, but there's still no effective treatments and there's no cure. So we are using mouse models to determine the impact of mutant Huntington on the brain, and particularly on the striatum and the cortex. And you can see this selective degeneration of the striatum here, and the caudate and the putamen in the normal brain, and how it's really degenerated in the Huntington brain. So in human disease, the age of onset is pretty much determined by the appearance of motor signs in involuntary motion like chorea. But degeneration occurs many years earlier, and the disease slowly progresses. In mouse models, there's a nice replication of some of the features in people with synaptic changes very early on and cognitive deficits, and then striatal volume decrease and cortical volume decrease. And also there's a progressive deficit in motor learning and in motor performance. So within the striatum, 95% of the cells are striatal projection neurons, which are GABAergic, and excitatory input comes in from the cortex and the thalamus. Our lab has been really interested in the mechanism of the very, very early changes in corticostriatal synapses, and we've used a lot of biochemistry and electrophysiology. So Lynn's done a lot of work with NMDA receptor trafficking and signaling postsynaptically. And we've also more recently started looking at synaptic plasticity and endocannabinoids and CB1 receptors, which I'll mention very briefly. But I'm going to be talking a little bit about glutamate release and uptake at these synapses, and then kind of describing how that led us into in vivo studies and into in vivo imaging, and combining these things with electrophysiology. So here's a diagram of one of Lynn's reviews looking at a postsynaptic spine in the striatum, and mutant Huntington expression leads to a destabilization of the synapse and altered trafficking of NMDA receptors to extrasynaptic sites. where their signaling, in contrast to the synaptic NMD receptors, is really promotes apoptosis and cell death. So you heard a little bit ago about the glue sniffer reporter, and we've started using that as well. So here, in collaboration with Tim and Mathieu Vanny, our postdoc Matt Parsons, who is now a professor at Memorial in Newfoundland, he looked at glutamate release in acute brain slices and stimulated within the striatum. And what he found was unexpected. So a lot of previous studies had looked at synaptosomes or dissociated cells where they saw a decrease in the clearance of glutamate and the expression of the transporter GLT1. But in a slice using glue sniffer, that's not what Matt saw. And he showed the clearance was pretty much the same, not only in the YAC128 model, but also the R62 model, suggesting that the uptake wasn't really different. Although he did find at very late disease stage that there was a decrease in the release of glutamate, but still no change in the uptake. And this is interesting and different from the previous studies because this is a brain with a semi-intact network, even though it's ex vivo, and there are astrocytes, and they're obviously very involved in clearing glutamate from the tissue. Ellen, who will be talking in a little bit, has sort of continued these studies using glue sniffer in a slice and stimulating. And one of the things that she found was an attenuation of long-term depression. So we get this in the wild type animals, in the wild type striatum, but it's attenuated in the YAC128 model. And this is also something that we saw looking at it with the electrophysiology by field potentials and also whole cell patch clamp. We see an attenuation or blocking of this LTD in YAC128 and also in the Q175 model of Huntington's disease. And there was a lot of interest in medical marijuana, but our results really suggest that in the case of Huntington's disease, THC as a direct agonist would not help motor symptoms at all, and it would actually interfere with synaptic plasticity. So we're kind of going on to work on enhancing endogenous signaling at the CB1 receptor, and we're starting to use the new sensor, BRAB ECV, to look directly at endocannabinoid release ex vivo and in vivo. Ellen's also been using the dopamine detection with D-Light. And interestingly, like we were talking a minute ago about glue sniffer, and it is very fast. It decays very rapidly, and the signal you can see with dopamine lasts a lot longer and spreads wider. So Huntington's disease being a motor disease, this study really shows the difference in a reaching task with the uncontrolled excessive movements with this severe Huntington's disease. So when we're testing motors in mice, we do look at their behavior with a rotor rod, which Ellen will be talking about, and also behavior in open field. And we look at forelimb movements with a lever pulling task. So Cameron Woodard, who recently graduated, was using a home cage system where the mice could be group housed, and they would go into this chamber and pull a lever to get water hundreds of times in a day, and they weren't interfered with in any way. So what he found was wild type mice really learned to do this fairly quickly and well, and while they did that, the variability in their pulse changed over time and decreased. And our Q175 mice, they didn't. So their rewarded trials stayed low, and the variability in their motion didn't really change over time. So after training, we took acute brain slices and we recorded in the striatum, and we found that there is a difference between the right striatum and the left striatum, where the left striatum, which is contralateral to the pod that they're using, actually has a smaller EPSC amplitude in spontaneous events compared to the right striatum, so which is ipsilateral to their pod that they're using. And Cameron did the comparison, and he found actually that this the size of the events on the left side here was really correlated with the variability of their motion. So moving on to the cortex really quickly, these are studies, experiments that were started by Majid Mahajarani and Alan Chan, and looking at the stimulation, sensory stimulation, and how responses spread across the cortex. And when they were looking in the YAC128 model of Huntington disease, they found that there was a bigger response, it spread wider. And when we did the same experiment with intracortical tetrodes, we found the same thing, where there was really big response, and it wasn't only in limb sensory regions, but it also was in the barrel cortex and also in the motor cortex. And looking at slices ex vivo, we found this surprising events that were NMDA mediated in the YAC128 mice, which we only really saw in the wild type mice if we induce glutamate spillover. So that was an interesting finding. So very briefly at the end, because I'm going to run through this, we have a talented new graduate student, and he is working with Tim, and he's looking at the cortex and the striatum simultaneously. So these mice express GCAMP in excitatory neurons in the cortex, and using mesoscale imaging, we can look at cortical activities and motifs that occur in spontaneous activity. But at the same time, Daniel is reporting presynaptic axons in the striatum with vibrophotometry. And he's done careful controls to show that the signals are independent, and stimulating the cortex isn't activating the GCAMP in the striatum. But there is a correlation between the axons in different areas of the cortex, and he can use this to map the areas that are correlated between where the fiber is in the striatum and what's happening in the cortex, and he can relate this to behavioral videos. So when there's motion in the video, how does that relate to the cortical mesoscale activity, and also how does that relate to what's happening in the striatum? So just finally, I want to thank too many people, which would take a whole 10 minutes. So I'll just have to say that Lynn is really amazing, and she's always has time for us, no matter how busy she gets. And Tim is very patient with us, and we've really, really benefited from having him around. And with that, I'll let Ellen take over and show what she's been doing.
Thank you very much, Maria. Yeah, so let's move on. I think we will keep question at the end of the session, if you don't mind, because again, the time is running. So let's move on with the next talk by Ellen Koch. Let me know if I mispronounce your name. I'm sorry.
It's Ellen Koch, but people always mispronounce it, so it's okay.
I'm sorry. So you can start sharing your screen.
Sure. Okay. All right. So yeah, I'm Ellen Koch, and I'm a PhD candidate in Lynn Raymond's lab. And Maria gave a really great overview of the many different projects, past and present, that have been going on at the lab. And now I'm going to talk about a project that I've been working on using fiber photometry in Huntington's disease mouse models. So fiber photometry is a technique that allows us to image the activity of fluorescent reporters in vivo while mice engage in behavior. So we'll start with a rodent that expresses a fluorescent reporter in the brain area of interest. Commonly used is GCamp, the popular calcium sensor. And these animals are then implanted with a small optical fiber. So in this case, we're implanting the fiber in the dorsolateral striatum. And this optical fiber sends light from an LED to the brain to excite GCamp in this case, as well as collecting the emission from GCamp and sending it back to a photodetector. And then eventually what you get is your signal, like a trace similar to the one that you see here. So here's a video of one of my experiments with a mouse doing the open field. And I'm recording GCamp activity in the dorsolateral striatum at the same time. So this is the signal, an example of a signal you would get. And then this at the bottom is the isospecific signal that we also get, which we use as a control. So when you excite GCamp around its isospecific point, in our case, we excited at 405 nanometers, you get the GCamp signal that's not dependent on changes in calcium. And so this signal isn't influenced by changes in calcium influx to the cells. So you can use this as a control. We use this to control for motion artifacts and bleaching, and we also use it in our calculation of the change in fluorescence. And then at the bottom here is an example of a mouse doing the motor learning task, accelerating rotarod, and an example trace of the GCamp signal before, during, and after a rotarod trial for this mouse. So for my experiments that I'm showing today, I'm using the YAC128 mouse model, which is a transgenic model of Huntington's disease that expresses a human mutant Huntington transgene with 128 CAG repeats. And I have two age groups. One is an early stage group. So this is 2 to 3 month old mice. They don't show a lot of behavioral differences yet. Based on previous research, but there are already changes happening neurologically, and then 6 to 7 month fully manifest stage. And then I express GCamp in the striatum, specifically in the dorsolateral striatum, under a synapsin promoter. So it's in the case of these experiments, it's just non-specifically expressed in neurons. It's not specific to a certain neuron type, but just in neurons in the striatum. So first we do the viral injection, and then we also implant the fiber at the same time. And then a few weeks later, we give, after the mice have recovered and there's expression of the virus, we habituate the mice so that they are comfortable being attached to the system and behaving. And then we do experiments. So I did open field at accelerating rotarod for 4 days. And after this, we check the fiber placement. And sometimes we also do slice experiments, such as plasticity experiments. So I'll start by talking about the rotarod. So for this task, the mice are on a rotating rod and they're trying to stay on, and it's slowly increasing in speed from 5 to 40 rotations per minute over 5 minutes. And they do 4 days with 3 trials per day for 12 trials total. And the most common measurement that is taken is the latency to fall. So this is a test of motor coordination as well as learning over the trials. And here's an example from a wild type mouse showing the GCamp activity in the brain on one trial on day 1 compared to day 4. And one thing you'll obviously notice, so the time on the rotarod is in red here, and before and after they are in an empty cage, just freely moving around. So one thing you'll notice is that they're on the rotarod for much longer by day 4 compared to day 1. And also the activity is elevated. When they go onto the rotarod, there's this overall elevation in the activity in the striatum on day 1 for the entire duration that they're on the rotarod. By day 4, you still see this elevation in activity, but then the overall activity actually decreases and becomes the steady activity that you see with these smaller amplitude peaks. And so this is something we really consistently see in our wild type animals. So as the mouse is improving on the rotarod, the latency to fall increases. And looking at the Z-score, so a measure of the average GCamp activity while they're on the rotarod, this decreases as they get better at the task as well. So there's this inverse correlation between the two. And interestingly, in my younger age group, which I'll focus on for the rotarod data, there was actually no difference when you look at latency to fall. So the Huntington's mice performed just as well as wild type. And if you look at the correlation between the latency to fall and the Z-score, so the GCamp activity, there's a very strong inverse correlation in wild type. But in the YAC128 mice, they actually start out with lower activity in the dorsal striatum on the first day. And then if you take all of the trials and look at the correlation, there's still an inverse correlation, but it's significantly weaker in the YAC128 mice. So even though they look like they're performing just as well, there is this difference in the activity in the striatum that's occurring. And so now I was interested in looking more closely at this behavior of the mice on the rotarod. And this was because one, while I was doing the experiments, I noticed some differences in the coordination of the mice, even though they didn't fall off. And two, because I wanted to see if there were other changes in the mouse's behavior that might explain the differences in the striatal activity that I was seeing. So here's an example of a mouse doing the rotarod. And I used the machine learning program DeepLabCut to track the position of the paws and some other points on the mouse while they're doing this task. And I wanted to look at how the paw kinematics change while they learn this task. So looking at this graph here with time on the bottom here, the two lines represent the top and the bottom of the rotarod. And you can see the red and green are the right and left paws. As the rotarod gets faster over the trial, the steps that they're taking get much larger. And they also have more of what I call paw slips, where the position of the paw actually goes below the bottom of the rotarod. And here's a comparison of a wild type and a YAC128 example. And you can see from the video, as well as these traces, that the YAC128 mouse is having more of these slips that are occurring and is overall more uncoordinated on the rotarod. So I quantified the number of foot slips, and I found that even though, again, these mice are staying on the rotarod for similar amounts of time, the YAC128 mouse is slipping, the mice are slipping more than the wild type mice with YAC128 in red here. And focusing on day 3, where I saw the largest difference, you can see that the with each trial shown here, there's more foot slips in the YAC128. And on this last graph, I'm showing the cumulative foot slips over time, and you can see how they separate around 150 seconds here. And the YAC128 have much more foot slips. So then I wanted to look at what GCamp activity looks like when these foot slips are occurring. And this is very preliminary data. I'm still working on this analysis, but I have some data from some example wild type animals so far. So what I did is I took every time that the mice show one of these foot slips, and I took ones that are at least half a centimeter, their foot goes at least half a centimeter below the rotarod, and I lined them up with the red line here is all of these foot slips lined up, and this is the GCamp activity before and after for many foot slips averaged together. And the one thing I really noticed is that right after the foot slip happens, there is this increase in activity, which would be when the mouse is kind of trying to reestablish their balance on the rotarod. And it is stronger, it looks stronger, at least in some of the example mice so far, in the left paw versus the right paw, which makes sense too, because we're actually imaging in the right striatum. And we're looking at, so this is the contralateral side. And you don't see the same pattern in the random control. And here's another example mouse. In this case, you see a little bit of a dip in the signal right when the slip occurs. And then again, we see this increase in activity after the slip. In this case, in both left and right paw. So the last thing with these experiments I'll talk about is a little bit of the open field results. So for open field, we have the mice explore an open field for 10 minutes a day for 3 to 4 days. And here's just an example of the activity in the brain, in the striatum at the same time as the video, and as well as the tracking with DeepLabCut. And as far as behavior goes, looking at the total distance traveled and habituation, which is a measure of their memory of the open field, there was no difference found in the 2 to 3 month old age group. In the older mice at 6 to 7 months, I did find a deficit in open field habituation, indicating a memory deficit. But interestingly, I looked at the peak frequency. So the frequency of these calcium events that occur during the 10 minutes that they're in the open field. And I found that the 2 to 3 month old mice have an increased peak frequency. And then really what I was really interested in is I looked at the correlation between peak frequency and total distance traveled. And I found that there's a correlation in wild type at both ages. So as they're traveling more, there's more calcium events. But in YAC128, there's actually no correlation. So again, this is showing disrupted striatal activity, even though their behavior is not necessarily different, or at least at the younger age, it's not. And then last slide, I just wanted to mention a few other experiments in the works. So I'm working on some experiments using 2-color photometry and taking advantage of the Cree-Lock system to specifically express GCAMP and RCAMP in different populations in the striatum. So I'm expressing GCAMP in the D1 pathway, the D1 spiny projection neurons in the striatum, and then RCAMP in the D2, the indirect pathway. So those are experiments that will show us how those two populations are affected in vivo in Huntington's disease. And I'll talk about glue sniffer quickly again. So I'm also using the glue sniffer glutamate sensor in vivo to see how glutamate signaling is affected during this task. And I just would quickly like to thank everybody on this page and more people, especially my supervisor, Dr. Raymond, as well as Maria, who really helped with getting this project off the ground. And then members of the Murphy Lab and actually JD Bain's lab in Calgary and the funding sources. And thank you to everyone for listening.
Thank you very much, Ellen. That was a great talk. Now it's time for questions. You can either write them in the chat or turn on your microphone and ask them directly. Maybe I will start. You stole my questions about D1 and D2 and how they might be affected. So I'm really happy that you plan to assess, different response you should get. About this, maybe do you plan to use these D1 Cree, D2 Cree animals to assess other change that might occur differently, like what Maria presented before, or do you plan to use them for your motor task?
So I guess both. So I am using them for the same task that I showed, so with the rotor rod and the open field. And then I've done a little bit of slice work too, like looking at glutamate release onto the D2 neurons as well. And in Huntington's disease, the D2 neurons are affected earlier on. The indirect pathway is affected first and then the direct pathway later. So yeah, I guess in both ways I'm looking at it. Right now I'm doing the in vivo work to start.
Okay.
Yeah, it's actually pretty interesting because in Huntington's disease, there's this imbalance of the two pathways in the striatum. And if we can figure out what's going on in vivo, we can actually try and reestablish that balance with neurofeedback. And this is something that we're looking into right now. And we're thinking about doing. We'll see how effective it is anyways.
Thank you. Any other questions?
Maybe a quick, Eric has one.
Yeah, so it's for a question for Ellen. Is exercise in the housing cage can rescue part of the yak mouse on the rod?
I'm just want to clarify the question. So like if the mouse have extra exercise,
yes,
okay. Yeah, that's an interesting point. Yeah, I'm not sure. yeah, I haven't really thought about that. I know there have been some studies that have done like wheel running in Huntington's models and have shown that it helped with some of the neurological changes. So, and I know, yeah, sometimes we do put wheels in the cage to help to kind of get the mice used to using wheels. Like that's not my experiments, but other experiments. So that's actually an interesting point that could perhaps having the practice in the cage could help with the foot slips that are happening on the rotarod.
Okay, I have maybe another question for Maria. So you talk about glutamate accumulation in the stratum and the role of the glutamate transporter. So we just heard about astrocytes. So do you think that they might be involved in that? And
absolutely, I really do. And so in different Huntington models, they found that astrocytes are more reactive in the striatum there. And we've also seen some changes with NMDA signaling that we actually think are due to glutamate release from astrocytes. So that is it's definitely involved in the response, even though they're seem to be equally as good at taking up glutamate. They're definitely the way astrocytes communicate with neurons in Huntington's disease is really different.
thank you. So if there's no more questions, we will close this session by thanking all the speakers. So Jean-Bastien, Keith, Maria, and Hélène, thank you very much for your great talk. It was excellent. And then we will have a short break, right, Mario?
Yes, absolutely. A short break, and we come back at 5:15 with the panel discussion.
All right. Thank you.
Thanks, everyone, for holding on until the end with this last session. As you saw, today was a very stimulating day with tons of approaches. We're using all the tools that optogenetics and opening for our science. And in the spirit of this Canadian Neurophotonics Platform, we wanted to finish our session today with a discussion on this whole concept of open science, which is particularly pertinent with the efforts that we're leading, which is really to try to enable and broaden the use of new hardware, new wetware, especially with the optogenetics and vectorology, but also all new software to analyze all of this data that we have. We've seen some very eloquent examples today of what people can do in the analysis space as well. So this is at the core of the objective of the platform. It's also something that Brain Canada, which is the main funder for the platform right now, is very is championing to try to develop more of an open science approach to the way we work. The platform is really trying to do this by linking all of these sites to work together to accelerate the development, give feedback, fastest feedback possible, to allow the developers to develop, to improve the development of the tools. And then, of course, a very important aspect is going to be to share all of this information to really help the community optimize the community. So we've sort of framed this last session with 4 speakers and discussing with them in preparing the meeting. There's a lot of great topics that came up in our discussions that come up all the time, with also a lot of challenges with open science. Of course, there are great things that are going towards a sharing approach, great things that it opens, of course, but there are also challenges. I list a few of them here that I think will come back in the discussions because they will be addressed in a different perspective from our speakers.
the need for the infrastructure to support open science, sharing and translating, all this data that we have. You saw it's so heterogeneous, especially in the interface of electrophysiology and cellular imaging. How can we work with these different formats, all the different software that people are developing? When should IP come in? Is it a barrier? Is it something that's enabling? Is it necessary? Is it a bad thing? What not? How do we get people to share ideas upstream of so that we can avoid just working in our corners and reinventing the wheel on each side, and eventually, ultimately, delaying research. Then comes the whole issue of credit assignment. If we're sharing all the data, if we're sharing ideas and so on, how do we properly assign credit to people? Which goes with how do we evaluate people's performance properly? And then how is that affecting the funding scheme? Funding scheme affected both ways, how we evaluate people, but also should this funding scheme be structured to support an open science? So we have 4 speakers. Adrien Perrache from McGill is going to tell us about neurodata without border, one aspect about data sharing, and the challenges of converting the data to and from common languages. Jeff Ledoux, who started a great initiative that's called Data Binge to stimulate sharing ideas, the problems and approaches. Marie-Ève, who will tell us a bit more about our Optogenetics and Vectorology platform. Again, how do we share info on tools, their performance, their development, and also this whole idea of trying to develop IP-free tools in this complex ecosystem. And we conclude with Ted Fong from the Neuro. We know that the Neuro has engaged very actively in a real, openly declared open science approach. And he can tell us a lot of their thought, the thinking process behind, and some of the interesting aspects of Boily. So this is for my introduction. I'll stop sharing, and we can start with that.
Hello, everyone. So thank you for having me. It's a real pleasure, and especially to talk about these topics that unfortunately we don't talk too often about, and we usually talk too much about science and not about like some of what I think are even more important questions when it comes to developing, future research. So today I'm gonna talk. Sorry. So is it working? Do you see the slide? Can you hear me? Great.
Yes.
So today we had like a lot of great examples of all sort of data that can be collected, especially with imaging. And it's not like big news to you that we're facing right now an explosion of data across different modalities, right? In electrophysiology, in calcium imaging, and many other techniques that are actually under development now. So this has been, of course, one major problem for years now. And many people, and I was like really lucky to be part of this, started to think about these problems a few years ago, and there was this initiative called the Neurodata Without Border that was started. It was at a meeting at Genevieve Farms, Howard Hughes Medical Institute, about like 2013 or 14. So what do I mean by that? So here are like very recent examples of the kind of data, massive data set that are collected. For example, with multiple neuropixel probes in vivo in mice, we're now talking about recording thousands of neurons simultaneously together with very precise behavioral data, with precise sensory stimuli, a lot of different modalities, so meaning different kind of time series that need to be synchronized and stored efficiently. Here, this is another graph that I really actually like. It shows some kind of the equivalent of the Moore's law for computers, but now in terms of the number of how the number of neurons that can be recorded with electrophysiology and how it progresses, over the years. And we see on a logarithmic scale that it doubles extremely fast. And so we're gonna be facing these issues of data explosion over and over, and we need to find a way of solving this problem. So what are exactly the challenges? I think there are three. Of course, we can talk more after during the open discussion, but let me focus on these three ones. The first, even talking about open science, even before talking about open science, it's about like what is going on within a lab. You have trainees coming in and then unfortunately leaving their recorded data set. They stored in some formats. And the problem is to make sure to ensure that these data are back compatible, because these are usually extremely valuable data, and you want someone coming in the lab a few years back, or someone else asking you for collaboration, to be able to read this data in a very efficient manner. Okay, so we need back compatibility. However, it's complicated because supporting such database requires technical expertise, and it's expensive though. So because most labs cannot afford to pay someone just to take care of the data. Of course, the second point, which is more related to open science, is that when you record such large data sets, you do not have the time nor the workforce to usually ask all the possible questions that can be asked with this data set. So it has become necessary to share this data and make them publicly available so that everybody can, analyze this data and add their own question with some of the data you've recorded yourself. And third, the problem with all this is that if we want these tools to be adopted, we need to also develop tools and software for processing, for visualization that are compatible across, these different labs using the same format. And as I said, wrote here the last line, a format is adopted only if people are provided with the tools to use it. Okay, so it's not about just having one format. We need to basically be a working format. And there is a beautiful example about this. It's astronomy. In the early 80s, so 40 years ago, they introduced the so-called feed format. And that was they were facing the same problem. That was the time of their first big, telescopes and satellites, and they developed a single data format so that they could share data efficiently and work very efficiently in small teams on the same format, and develop also tools. So what is amazing about this is that this format is still used today, 40 years after. The same format, it has barely evolved. Even if it's not totally optimal, it's still, good enough. It's supported in all possible coding language you can think of. There are APIs in C, Python, and even the most modern languages. And it's readable by many software, including ImageG, Photoshop, even GIMP can read the feeds format. So to tell you, it's extremely broad, very useful, and very efficient. The joke is that the European Union, about 10 years ago, developed a new satellite. They spent between 10 and 20 million euros to develop a new file format for this new kind of data, and no one is using it, all right? Because there was this one that everybody loved and they kept it. So when it comes to data, especially in neuroscience, but it's the same as in astronomy, we're now seeing that there is some sort of split, but it's actually very positive to me that there is some data providers, centers or labs that collect very valuable data, and data users, people that actually do not necessarily run experiments, but we can use these data to ask the question they really want to address. And here we know this is a famous example that you have this large collaboration across many different astronomy centers around the world, and almost one postdoc at the MIT will analyze this extremely large data set coming up with the image of a black hole, right? So, and this is only possible because all these data were formatted exactly in the same way, and everything, all the platform and infrastructure were already in place. So as I said in the introduction, the idea is that can we do it for neuroscience? So that's the goal of the Neurodata Without Borders data format. Here, this was a seminal paper in 2015, and now it has actually even been improved in many different ways by many teams, especially in the US. And the idea is the following. You have one data format, one file, it could be actually several, it doesn't really matter, in which you basically encompass all the different data you have, which include, of course, all the identification of the animals, extremely important, you know, for long-term tracking of what the experiment was, the design of the experiment, of course, the data, how it was acquired, the data themselves across different modalities. It doesn't matter, it could be calcium imaging, could be electrophysiology, and also behavior, whatever the behavior is, okay? And it's versatile. You can add whatever you like, okay? It's not rigid, it's very flexible. And there are APIs that have been already developed in MATLAB, Python, and other languages. It's supported by many different big institutes, especially the Calvi Foundation, the Allen Brain, in the Brain Initiative. So then you need to store this data. There's also a lot of initiatives going on. I don't need to spend too much time about it, but it's also important is that you don't necessarily share the data only when you're asked to share the data, it's that you put the data online, but it requires also a lot of infrastructure actually. So how do we make people adopt it? Well, we need fund because actually it costs money to adopt or to reformat your own data in your lab. So these are many mechanisms, but not too much actually here in Canada, unfortunately, right now. You need to offer technical assistance to the labs to basically help the labs to readapt their infrastructure in terms of APIs, how the data are stored, how the data are read and processed. We need to also develop more software and update this existing softwares. So to finish, you know, shamelessly doing some advertisement about things that exist and are going on. For example, here at the Neuro, like a software like Brainstorm that was originally developed for the MEG and EEG analysis now is capable of doing a lot of different things. It's open source, and now basically NWB has been adopted for the software and can be used natively. And especially, for example, for multi-channel electrophysiology, we worked with this past student at the Neuro, Konstantinos Naziotis, and that was an example of like everything that can be made directly. So we have a tool that natively read from this format and that can process the data how you like. He now actually has his own company, Nevronas, whose goal is to actually go in the labs and do all this work for the labs. And last, very quickly, again, shamelessly advertising at the Neuro, we launched a couple of years ago the Open Science in Action prizes. Stay tuned for the announcement of the 2021 prizes. I strongly encourage, especially trainees, there will be like up to, I think, 7 prizes for trainees. So whatever, if you have new ideas about data, how to store data, how to process it in open science, even your devices, this is the time. We're here to hear for all the crazy ideas you could have. So thank you, and looking forward to the discussion we'll have later.
Thanks, Ali. Great. It sets the table well for this further discussion on data and conversion and sharing. The idea today is we're having the 4 speakers talk briefly about make their pitch, 10-minute talks and so on, and so that we reserve the end of the session for discussion, an open discussion on all these topics. So you can certainly start posting your questions, but we'll get back to them at the end of the 4 speakers. Go ahead, Jeff.
Can you guys hear me?
Yeah, we see your question.
Okay. Yes. Okay, so my name is Jeff Ledoux. I'm a staff member at the Center for Brain Health at UBC, and I work closely with Professor Murphy on these two entities here, the Dynamic Brain Circuits Research Cluster and the NINC, which is our Imaging and Computation core facility. And these have given rise to something that we call the Data Binge Forum, which I am going to tell you about in the next few minutes. So this being the open science discussion, I wanted to start with a definition because it can mean many things to many people with the emphasis placed on different aspects depending on the conversation. So I like this definition that is centered around the researcher and emphasizes reproducibility. It goes, scientific research conducted and communicated in an honest, accessible, and transparent way such that independent researchers can reproduce the results. So this suggests a critical aspect of open science and just science in general, in that it is the constructive criticism from and interactions with the independent researchers that make science work. So we must find effective ways to interact to gain feedback, suggestions, alternate ideas, and collaborations early on in the discovery process. And this is what we've been trying to do with the Data Binge Forum at UBC. So before I get into the nuts and bolts of Data Binge, I just wanted to put up this diagram to show the kind of collaborative data analysis model we've been promoting at our center. So on the left here, we have different streams of data that might be of interest for a particular project. We all tend to be experts on a particular type of data as we spend time in particular lab learning techniques, but we want to get the most out of our data. So extracting meaning from this data is incredibly important. And we feel this is best done as a group, leveraging all the expertise in our community. And this is where Data Binge fits into the process in this middle loop. And the hope would be that the output of all of this would be more open collaborative papers with accompanying open data resources and analysis pipelines freely available on GitHub for people to download. So the Data Binge meeting as it stands now is typically a drop-in meeting on Zoom. Everyone is welcome to come with their questions, their ideas, and their expertise. The meeting is facilitated by our graduate student peer tutors and our undergraduate open science specialist. And I'll say more about them in a minute, along with myself and Professor Murphy and other faculty who come to the meeting. So at this meeting, we try to match the expertise in the group with the problems that are coming to us. We create a project channel on our Slack workspace to facilitate interactions between our Zoom meetings. And we encourage people to share the data needed to address their problems on open science framework projects and post the code that they've been working on our GitHub organization. So at the moment, this is just a snapshot of our Slack workspace. You can see a number of the project channels that we've created to work on things with different labs. We also have these help channels for particular languages or topics that people might be getting tripped up on. At present, we have 231 members in this workspace, more than 50 project channels, and a lot of messaging activity going on. In 2020, we saw 12 papers across 9 different labs that were directly supported by our efforts at Data Binge, along with the creation of 51 projects on Open Science Framework, of which 11 were made public. Data Binge has helped over the years support over 130 trainees in learning to code. And all of this is facilitated by our neurodata peer tutors and open science specialists. So the current group of tutors that we have assembled at UBC are these individuals. There's Abhijit, Pankaj, Peter, Adrian, Nick, and Jordan. They have an incredible breadth of experience working with different types of data and analysis problems. I'll post this link at the bottom in the chat after I'm done speaking, so you guys can take a look at the particular skill sets that they have if you're interested. But I just wanted to say a few words about how we chose these individuals. So early on in 2020, we had an event called the Brain Circuits Coding Challenge. The challenges that people put forward came from different labs at UBC to illustrate some common workflow in their lab or a current research question that they had yet to solve. We had 6 such challenges from 5 different labs and 17 people participating in the challenge. We hosted the challenge materials on Open Science Framework so we could reuse them later. And the solutions that were submitted after the coding challenge were part of the evaluation to become one of our peer tutors, along with a CV and personal statement. I also wanted to spend a few minutes describing our undergraduate open science specialists. So these are co-op students that do a placement with us for 4 or for 8 months. They typically come from physics, engineering physics, biophysics, biology, computer science. And these individuals have really been essential in supporting the nuts and bolts aspects of helping researchers adopt open science practices, like by making use of tools such as Open Science Framework and GitHub and Compute Canada systems, other UBC resources. They're extremely talented and productive. They've made a lot of contributions to things such as our data management and data sharing white paper, our workshops, internal guides that we've made for the Compute Canada resource allocation competition and sharing data on Further. There's just a couple of examples I posted here. Keegan is the student currently in this role. This is a video that he posted on our YouTube channel after our statistics workshop where he's explaining how to do resampling in R. And prior to Keegan, we had Ashley, and she's posted a video on our YouTube channel explaining a particular clustering algorithm that had come up many, many times at DataBinge. So is all of this leading to more open outputs? I think it might be. So we've been seeing more resources published alongside papers beyond what you would normally put on a journal website as supplementary material. I put a couple examples here. These are one project called PyDose from the Raymond Lab, which is a home-built system for delivering particular dose of drug to an animal in its home cage. So this project has all of the code and instructions for building and the files you would need to 3D print the parts. And the synthetic animated mouse project, this is one that came from the Murphy Lab last year, and it contains all of the data needed to reproduce the synthetic animated mouse on this Open Science Framework project. So at this point, we really see DataBinge as underpinning all of our larger collaborative efforts, including our new CFI project, our involvement with the Canadian Neurophotonics Platform, and our new collaboration with the University of Washington Computational Neuroscience Center. We'd like to keep DataBinge online, but that I think may be difficult with everyone trying to get back to normal or something in the fall. But I will say that we believe this model is scalable, although the choice of platform is kind of important for privacy reasons and satisfying university policies, as well as maybe easily giving us metrics that we can track for our own sake in terms of measuring our progress. So with that, I'd like to thank our UBC VPRI. It's their support of the Brain Circuits cluster that has made this possible, and thank all the Brain Circuit team members, the tutors and undergraduate students, as well as Tim Murphy, the cluster lead, and thank you for your attention.
Great, thanks, Jeff. While you stop sharing and Marie-Ève sets up, just a word of logistics. I encourage people to put their questions on the Q&A panel rather than in the chat. And be careful, people, when you respond to the chat. If the question is posted to everyone, respond to everyone. Like Arthur Kanya just asked a question about how expandable it is to other fields, not just electrophysiology, but behavior, imaging, and so on. And Adrien, you responded it's good for everything possible on Earth, but you just said that to the panelists, so Arthur didn't get your answer. So be careful about your the forum that you use. And as I said, we will come back to questions. I have questions for Jeff, but and I'm sure you guys have, but let's listen to Marie-Ève first, and then Ted, and we'll come back to questions. Marie-Ève?
Yes, sorry, I think it's, oh, can you see the screen?
Yes, perfect.
Okay, good. So, okay. So I'm going to talk about the Optogenetics and Vectorology Foundry that Yves introduced at the beginning. And basically this, this foundry is involved in tools development, resource sharing, and you know, my group in particular is interested in developing open source AAV capsids. Oops. So Yves presented this this foundry model on which is based the Brain Canada funding that we got last year. So it's basically an open approach to development and optimization of tools. And the idea of the BioFoundry is basically a cycle, an optimization cycle, which begins with the design, and then you build something, and then you test it, and you know, with the results from the test, then you go back to the design stage and hopefully go through the loop a few times, and then eventually your new tool, which is optimized, comes out of the loop and is being distributed. And this is definitely an open science or open source model, or fits with the open source ideas. You know, through the design phase for us involves developing or designing new genetically encoded light sensitive tools, like sensors, for example, of different kinds. And this is, most of it is a work from Robert Campbell's lab. And already after, you know, this tool is developed, there's possibility of distributing the tools through plasmids. There's us, you know, my group is the viral vector core of this cycle. So the building group. So we develop, you know, the configuration of the virus. We can help you choose the preferred capsid, and then we can package it in different configuration, and then we send it to the testing nodes. And so we are packaging, I just want to say that we're packaging these new tools, but also packaging tools that are being developed by, you know, other developers that are not really involved necessarily directly in this group. So we are really involved in making a lot of the genetically encoded tools available to the community. And I believe that our model, which is a little bit different from some of the other platforms, is also, you know, facilitating a lot of people's research, in that we can help you, you know, develop custom, basically, tools for your own research. So we're distributing stuff across Canada and in labs around the world, and we have, oh, sorry. We have distribution agreements with different tools developers also. So, so then once we build, we send to these testing nodes. I'm going to describe that a little bit later. And we are hoping eventually, when this data comes out, to make all of this also available through different, you know, platforms such as the Primate Optogenetics Open Database, you know, and I'm going to talk about some of the other possibilities as well. So this loop basically involves a testing ecosystem. So the two cores, production cores, are important, but around that is a whole ecosystem of testing, in which basically there's a first line of testing with, maybe less complex, like cell preps, invertebrates, lower vertebrates. And some of those testing sites don't necessarily need, for example, the viral production. They can give rapid feedback about this new sensor, for example, without having to go through the viral production phase. Some of them do use viral vectors, but this is the first line of testing. And then there's second line of testing in slightly more complex animals and cell types like non-human primates, rodents, human cells, organoids, and most of these will require viral vector production. They can give feedback on the viral vector themselves, and they can also give feedback about the biosensors. And so a lot of labs, across the country are involved in this, in this testing ecosystem. And hopefully when new biosensors or actuators, when new tools come out, they can be deployed in the community. And, at the research level, hopefully there's some eventually translatability. This is why also we've now have more and more testing nodes that are closer to human, iPS cells, non-human primates, and so on. So the first, or basically to get this ecosystem to really work together, we have embarked on an initial project or benchmarking project in which we'll be using GCAM8 as a test case. And the idea is to basically put together a standardized pipeline of tests. The idea is to get some sort of quantitative protocols that can be used and ways of analyzing that can be used across different preparations, in vitro, in vivo, different cell types, as well as across species. So you saw that in the different testing sites, there's different species and cells and everything. And also this will be done considering the different experimental paradigms that the labs that are participating are using. So to do this requires also eventually will generate a lot of data, and this data will need to be shared. And we will use this NWB pipeline data format that Adrian described. And this in itself is actually also a project, because I believe that it's being used now in some groups like the Allen Brain Institute, but they're within the same institution. With this testing ecosystem, people are all over, and so this will be a challenge, but we think we have a great environment to do it. So I just want to now maybe move on a little bit to my main interest, since I'm the AAV person in the group, the building core basically. I'm going to tell you a little bit about the main viral vector that we are packaging in the lab. So it's the adeno-associated virus, the AAV, which is a small virus which has a linear single-stranded DNA genome of about 4.7 kb. And it has a very few genes, basically a rep and cap gene. And it's fairly easy to package these viral vectors in mammalian cells. And after it's being packaged, we need to purify it. And, even if the virus or the viral vector is in theory simple, it is made of quite a number of components. Basically, you need a promoter, you need a gene of interest, and there's the capsid of the vector itself. And so these AAVs are used as vehicles to bring the gene of interest. So for example, the genetically encoded sensor into cells and into animals. And we are always looking, in the community has been working really hard to develop better AAV-based transduction, and different strategies that have been used and are currently being used. So one could work on the promoters, for example, and this includes short cell-specific enhancers. And the promoters will basically target the expression. So the AAVs will bind to a variety of cell types. They do have tropism, but they're not that specific. So you might need a specific promoter if you want your gene of interest to be expressed in a particular cell type. The gene of interest themselves, can be worked on. The cassette configuration, so what's in there, is there a stabilizing sequence, the poly A, the orientation, and then all the sequences associated with the AAV itself. So the capsids, some sequences that could be involved in minimizing degradation, etc. And overall, even if it's a simple vehicle, you do have many, many parts that can come from a number of contributors. And this, considering that all these parts on their own are typically associated with intellectual property, this can generate an entanglements in terms of IP, and could potentially, slow things down and create barriers in the development. So we've basically decided to approach this with an open science, in mind. So we have been working on these open AAVs by identification of new capsids. So there are AAV serotypes, like numerous AAV serotypes that exist in nature that have different characteristics. Some of them might have lower pre-existing immunity. So we are looking for characteristics that are desirable. Most capsid development today is based on engineering of existing capsids associated with IP. So we are thinking that if we're finding new capsids in nature that with which there's no intellectual property associated, well, these ones can have new, interesting characteristics, but we could also work on them to modify and improve some of their characteristics. And the other thing with new AAV serotypes that we don't know what we're going to find. There could be other things that are not necessarily, in the capsid, but other stabilizing sequences that could be interesting for gene transfer, maybe outside of the cap gene. So there's potential there. So with the help of a research associate in the lab, Claude Lama, and a really great summer student, Alexandra Gravel-Nolin, we have established a collaboration with the zoo in Granby, which is closer to Sherbrooke. And Claude has been visiting the zoo to collect species from different animals, and has developed a protocol to enrich the sample with AAVs. So all kinds of different steps, and basically now has identified a new cap gene from a mandrill sample. And, we did some alignments with some of the existing AAV sequences and have found that compared to some of the sequences, there's an insertion there that is present, which is, not completely unique. It's also present in some of the other cap genes, but very interestingly, This has been associated with increased infectivity. So we are really looking forward to continuing the characterization of this new AAV. So we'll be packaging it or packaging viral vectors using this new cap gene. We think that we'll be using the same protocol because overall it's still a similar sequence, but we never know. We might have some optimization there to do. We are, of course, going to do some in vitro characterization with cell lines, human iPS cells, rodent primary cells. And of course, we are hoping that the testing sites from the Optogenetics and Vectorology Foundry are going to be helping, if they can, to do that. Same thing for in vivo characterization, although there's going to be different things to look for: tropism, infectivity, blood-brain barrier crossing, retrograde transport, transsynaptic transport, etc., etc. So we'll be looking for the testing sites for that. And we're looking also to characterize the full AAV sequence, not only the cap gene, because we never know what we might find there. And of course, this sequence and this capsid will be made readily available. We are not going to seek any IP early in the discovery process. And I'm throwing the question out there for the discussion later. I think that this will accelerate innovation and potentially facilitate therapy development. So of course, this is a team effort. So I want to thank all the members of the Optogenetics and Vectorology Foundry. I've put the names of the PIs there, but I would like to thank all the people from the different groups. The viral vector lab, and particularly I've shown some of the data from Claude and Alexandra, and also some collaborators and students of mine that are working on different things. And if you want to know what we're doing and what's available, we have the website. And I just want to, we're going to possibly discuss credit assignment. For those who didn't know, there is something called a research resource identification number. We have one. So if ever you're using viral vectors that we have prepared, you can use this number to cite us in your papers, and this actually helps us, track the people who are using this and the papers that are published with our viruses. So thank you very much.
Thanks very much, Marie-Ève. And the last speaker, Ted, is going to tell us about the neuro experience on the open science.
Okay, thanks. Thanks, Eve. Thanks, everyone. So let's see. Okay, hopefully you can see that. So yeah, this is, I guess, for the hardcore group who's still with us after, 5 hours of talks. So, the first, you heard earlier, two of my my trainees give talks about iPSCs and organoids, and I get the job of talking more about the social sciences part of what we do than the hardcore science, which is probably a good thing at this stage of the day. So, I'm going to tell, as Eve said, I guess I was asked to tell you a little bit about the experience at the Neuro in open science. And I like to put this first slide up because I think it really illustrates a lot of the things I'm going to try to convey today. And this is on a mural in one of the conference rooms at the Neuro. And seen here is Wilder Penfield, the founder of the Neuro. And he's flanked by Jasper and Cone. And what's interesting is that he's, put himself in the center of this mural with many very famous neuroscientists. There's Cushing, and Osler, and this is Charcot, Claude Bernard, Ramon y Cajal, and others. I mean, there's, you know, it's a who's who of neuroscience. And I guess what he was trying to show here is that, you know, the mission of a place like the Neuro, which is a hospital and a research institute, both for trying to cure or help people with brain diseases and also to understand the brain. And, you know, this is sort of what we've been trying to do. And in the past, especially I think in the past 10 years, is trying to put these two things together. And just for those of you who don't know the Neuro, this is a drawing that Penfield had. And the idea here is that the clinical and the research floors are intertwined. And we have a pretty, so you know, this is the kind of idea of having bench to bedside. And so we're a research institute of McGill University, and we're also a hospital of the MUHC, the McGill University Health Center. And we're, you know, it's very balanced. There's about 600 staff on each side. And, and a lot of these are trainees, but also scientists, physicians, neurologists, clinician scientists. So it's a really nice place to work because I think we very much strive to make this a reality. And, you know, we do this, we focus on a number of different programs, clinical programs, which are centered around different diseases, which are listed here, and research programs or research groups, again, which in many cases match up with some of the clinical groups, and that many people are part of both groups. And we've established over the last 10 years a number of platforms, and animal models platform, CBIG, which I'll talk about later, the early drug discovery unit I'll talk about later, and then the brain imaging and clinical research unit are infrastructures we've tried to build. But what I'm going to mostly talk about, and the theme of today's, of this current session about open science, and the institutional approach that we've taken, the stance that we've taken at the Neuro. And, you know, the first question one may ask, and I think obviously to this group, we're sort of preaching to the converted, but, you know, I think there are two main reasons. One is to accelerate our understanding of the brain. This is perhaps one of the biggest questions in science and society, is understanding how our brain works. And, you know, there's a number of challenges, including the challenge of, you know, just the scale of the problem and the reproducibility and the different, and you know, the speakers early that preceded me have addressed many of the issues here. But also for me as a neurologist, I think one of the reasons is to try to find better treatment to brain diseases. There's over 150 million people that suffer from brain diseases in North America alone. About $1 billion a year goes towards this. So it's a huge problem. And we have almost no treatment for most of the most devastating neurological diseases. So, you know, we feel like something has to be done. And open science, I think, is not going to solve everything, but I think it's one of the tools that I think could help accelerate things. And, you know, this is a quote from Henry Ford. And so if we just keep doing the same thing, you know, I don't think we're gonna get any results. So I think to try to change things is important. And this was started in 2016 through a very generous donation by Larry Tannenbaum, shown here with Guy Rouleau and Suzanne Fortier. And of course, you all know who this is. And so, you know, this was, what was very nice about this for me is that, I don't know, probably most of you know, Larry Tannenbaum is the owner of the Toronto Maple Leafs. So for him to give money to Montreal was a really nice thing for us. And so the idea was that we did this not just as a top-down, but as a bottom-up approach. There was about 1.5 years of consultation within the faculty at the Neuro, both clinical and research. And the goals were to increase the impact of research, to develop tools and infrastructures, which I'm going to discuss a little more, but also as a social science experiment in the sense to measure the outcome. Is it true that social science, that open science actually does accelerate and make things go faster? I mean, it gets to the question that Marie-Ève just asked, like, does IP slow things down? Well, or does open science accelerate things? And these are questions that we intuitively think are true, but we don't really know the answers to. And finally, and perhaps most importantly, the gift that the Tannenbaum gave us, really half of that gift goes towards proselytization, or to preaching and converting others towards this mission. So I'm going to just mention that at the end. And so after this initial consultation, we came up with 5 guiding principles. The first, which I think is almost now mundane, which is that data is released and research are released no later than publication. I think this is probably now, 5 years later, probably out of date. I mean, I think this is true in many places. But this is also true for all our partners, including industrial partners and all our pharmaceutical partners. Principle 3 is that patients are participants, and that to create the infrastructure to share data and resources from patients that we see in a very respectful way, respecting their privacy. And 4, no restrictive IP, including no patents, will be filed. And finally, because we're not a dictatorship, that we respect autonomy, and that we can't force anyone, even people in our own institution, to abide by these principles. But we've had overwhelming adherence and support for this. So what we're doing in practice, 5 basic initiatives: open biorepository, or CBIG, open early drug discovery, IP compatible with open science, open access, and open data. So I'm going to talk mostly about these 2, open biorepository and open drug discovery. So one of the things that we've done, and I think could be very interesting for the community here, is to set up a biorepository called CBIG, for Clinical, Biological, Imaging, and Genetic Repository. And what CBIG is a multi-dimensional. So it's not just a biobank where you have a tube in a freezer that just says, Alzheimer's on it, or it says epilepsy, but it has multi-dimensional data, which includes, on that same sample, it has not only the biosample, but brain imaging, clinical and neuropsychological data on the patient, and genetic data. And this is data that is then accessed. You can access both the data and the samples through our website, and this is now completely, it's online and uses the informatics platform of Alan Evans called Loris for data queries and for sample queries. So so after 3 years, over 24,000 samples, we have an ethical framework so that patients are consented, privacy and data security is guaranteed, multiple diseases, and we have now an initiative for healthy controls. The idea is to get 1,000 healthy controls into the biorepository. What I think is very interesting is that we have industrial partners, including large pharma, that are supporting this initiative, despite the fact that there's no IP, that this is all an open repository. So, and we have a number of institutions in Canada and internationally, and a number of initiatives such as Canadian Open Parkinson's Network, Capture ALS, etc., that are part of this initiative, and where CBIG serves as the main repository. So I'm thinking here that with the photonics initiative, this could be certainly, I think, a good resource for this community to access patient-derived material. The other thing you heard a little bit about, so I won't go into any of the details, but one of the main outputs of CBIG is to generate human samples. I mean, we've done, you know, the animal models are very powerful for certain questions, but for diseases, they've been rather disappointing in most cases. And so the idea now with this iPSC approach, and this is being spearheaded by Tom Durkin, my colleague, who runs this facility, the EDDU, the Early Drug Discovery Unit, to generate from just blood samples from CBIG and elsewhere, making human neurons, organoids. You heard V talk about this and Ghislaine about what we're doing with these organoids. And the idea is to take these samples, reprogram, and then develop not only basic research questions, but also drug screening and assays for screening and drug discovery and genome editing, CRISPR-based screening. So, and again, you know, the idea is that with patient samples, the idea is to try to get a better understanding from these organoids, brain organoids. We're using approaches such as single cell approaches, single cell RNA-seq, to try to understand how similar these organoids are to actual brains on a transcriptome level, but also, as you heard earlier, on the electrophysiological and functional level. And the idea is then to take, for example, organoids and then screen them with compounds, and compounds that correct phenotypes that we identify can then screen organoids from multiple patients, such that you will find some patients responding, and you can even go back then to the clinical picture and go back and stratify patients that have responded, because these are now live patients that we're following in the hospital. And so again, you know, the EDDU, as being one of the main platforms for the Tannenbaum Open Science Institute, we've secured significant funding beyond the gift of Larry Tannenbaum, and a third of this is from industry. In addition, we've had partnerships with entities such as the Structural Genomics Consortium. I mentioned earlier our partnership with different industry who have really embraced this approach. And we're even now in partnership with the SGC developing potential ways of drug development without IP. You know, just to conclude, as I said, I mentioned at the beginning, one of the main missions of TOSI, of the Tannenbaum Open Science Institute, is to promote this across Canada first, but then internationally as well. But people on this call are from a number of institutes in Canada, and the idea is that there's significant funding for projects and for commitments towards open science. Our goal would be to have sister institutions across Canada that would adopt a similar open science stance. It doesn't have to be a full, you know, university. We've done this, you know, as just an institute within McGill, but I can, you know, imagine that institutions like Cerveau or like, you know, at UBC, the Moafagian Center for Brain Health could take on similar positions. And I would say the same for, at Robarts, we have a project in common where we're adopting this position. So, I mean, I think that's it. So, you know, I just want to thank you for your attention, and happy to discuss these things further during the panel discussion.